Generally speaking, our central nervous system is made up of three parts: the cerebrum, the cerebellum, and the brainstem. The cerebrum consists of two nearly symmetrical halves called the cerebral hemispheres, and deep within these hemispheres lie the basal ganglia. On a coronal or axial section of the brain, the outermost area represents the cerebral cortex, which is made up of gray matter that consists of billions of neuronal cell bodies. The axons that are connected to these cell bodies create the white matter of the brain, which is the innermost area. One prominent white matter fiber bundle is the internal capsule, which is like a highway that allows signals, and thus information, to flow to and from the cerebral cortex. On both sides of the internal capsule, we can see areas of subcortical gray matter that form the basal ganglia. The basal ganglia are very important for providing a feedback mechanism to motor cortices for initiation and control of voluntary movements. So, for example, you want to write your name on a piece of paper. First, you plan the movements using your prefrontal cortex, and that sends a signal to the motor cortex as well as the basal ganglia. The basal ganglia now help the motor cortex prepare for and initiate the action. The result is that you position your arm so that you can start writing. Then, the basal ganglia ensure that your hand movements are as precise and executed as planned, and while doing so, they also maintain your posture. In addition to this, the basal ganglia can help you learn new procedural motor skills, like riding a bicycle. The basal ganglia are a collection of nuclei that include the caudate nucleus, the putamen, the globus pallidus externus, and the globus pallidus internus. These nuclei have highly complex connections with other parts of the central nervous system, like the cerebral cortex, the thalamus, and the brainstem. But the two most important structures that are closely related to the basal ganglia are the substantia nigra of the midbrain and the subthalamic nuclei of the diencephalon. Now let's look at each of these structures in detail. First up is the caudate nucleus, which is shaped like the letter C. The caudate nucleus can be divided into three parts: the head, the body, and the tail. It follows the lateral ventricle of the brain, which is medial to it, and curves around the thalamus. Laterally, the caudate nucleus is bounded by the internal capsule. On a transverse section of the brain, the head can be seen forming the lateral wall of the anterior horn of the lateral ventricle. On a coronal section, the body of the caudate can be seen along the wall of the body of the lateral ventricle. The tail of the caudate curves around the posterior end of the thalamus and follows the inferior horn of the lateral ventricle, forming its roof. The putamen and the globus pallidus externus and internus are sometimes collectively referred to as the lentiform or lenticular nucleus. This is a wedge-shaped nucleus with a wide base pointing laterally. On transverse and coronal sections of the brain, we can see that the internal capsule is medial to the lentiform nucleus and separates it from the caudate nucleus and the thalamus. Lateral to the lentiform nucleus is the external capsule. Thin sheets of white matter pass vertically through the lentiform nucleus, dividing it into its three components. First, the most medially positioned nucleus is the globus pallidus, which has a lighter appearance because it contains more myelinated nerve fibers. The globus pallidus is further divided by a sheet of white matter into the globus pallidus internus, lateral to the internal capsule, and the globus pallidus externus, which is medial to the putamen. The putamen is the most lateral of the lentiform nuclei and has a darker appearance. It's positioned lateral to the globus pallidus externus and medial to the external capsule. The putamen and the caudate nucleus are sometimes referred to as a corpus striatum, or just the striatum. These two nuclei are separated by the internal capsule, except anteriorly, where the head of the caudate nucleus is continuous with the putamen. Superior to this connection, small bridges of gray matter between these nuclei pass through the internal capsule, giving it a striated appearance, hence the name corpus striatum. Let's take a short break and see if you can identify the function of the basal ganglia, as well as the basal nuclei that comprise the basal ganglia. Let's switch gears now and talk about other nuclei that are closely related to the basal ganglia. Inside the midbrain, ventrally, between the tegmentum and the crus cerebri, there's a darkly pigmented area called the substantia nigra, which is Latin for black substance. It contains cells that store granules in their cytoplasm, filled with a pigment called neuromelanin, making this region look darker in color. The substantia nigra can be divided into two parts: the substantia nigra pars compacta, or SNPC, and the substantia nigra pars reticulata, or SNPR. SNPC contains dopaminergic neurons that project their axons to the corpus striatum, forming the nigrostriatal pathway where they release dopamine. Then there's the subthalamus, which is a part of the diencephalon located ventral to the thalamus, lateral to the hypothalamus, and medial to the internal capsule. It contains a subthalamic nuclei, or STN for short, which assists the basal ganglia in the regulation of movements. The STN receive afferent fibers mainly from the globus pallidus externus and project efferent fibers mainly to the globus pallidus internus. Now that we've covered the main basal nuclei, let's see how they work together to assist the motor cortex in control of movements. In order to pass messages between each other, neurons of the basal nuclei use different neurotransmitters. They either release excitatory neurotransmitters, like glutamate, or inhibitory neurotransmitters, like gamma-aminobutyric acid, or GABA for short. The basal ganglia don't have any direct connection with lower motor neurons, so they can't control the muscles directly. However, they influence the motor cortex through the thalamus. Basically, information enters the basal ganglia through the corpus striatum, and it leaves it through the globus pallidus internus, or GPI for short, which has an output on the thalamus. The information flow can follow two different pathways, the direct pathway and the indirect pathway. The normal connectivity of the direct pathway results in the excitation of the motor cortex, while the normal connectivity of the indirect pathway results in the inhibition of the motor cortex. To remember which pathway produces which effect, just recall that the indirect pathway inhibits the motor cortex. Now, to better understand how they function, let's dive into both of these pathways step by step. The direct pathway starts with the substantia nigra pars compacta, which contains neurons that project to the corpus striatum. These neurons release dopamine from their axons, which binds to D1 receptors found on the neurons of the corpus striatum. Neurons that have D1 receptors project directly to the GPI and inhibit it by releasing GABA. Now, normally, the GPI projects to the thalamus, where it releases GABA, thus inhibiting the thalamus and preventing it from releasing glutamate and stimulating the motor cortex. However, because the GPI is inhibited by the corpus striatum, the thalamus is then disinhibited and can stimulate the motor cortex, thereby assisting with the initiation and execution of the desired voluntary movements. At the same time the direct pathway is activated, the information passes through the indirect pathway as well. The indirect pathway also begins with the neurons of the SNPC that project to the corpus striatum, where they release dopamine. This time, dopamine binds to the D2 receptors, found on the neurons that project to the GPE first. Once activated, these neurons release GABA, which inhibits the globus pallidus externus, or GPE for short. Normally, the GPE projects to the subthalamic nuclei and releases GABA to inhibit it. This prevents the STN from releasing glutamate and stimulating the GPI. But because the GPE has been inhibited by the indirect pathway, the STN breaks free and starts releasing glutamate to stimulate the GPI. This excitation causes the GPI to release more GABA, which inhibits the thalamus and results in the inhibition of the motor cortex. Even though it might seem like the indirect pathway wants to sabotage the direct one, it actually helps the motor cortex by suppressing the unwanted muscle contraction while producing voluntary movements. Alright, as a quick recap, the basal ganglia regulate initiation and execution of movements, regulate posture, and assist in learning new motor skills. They're found deep within the white matter of the cerebrum and consist of the caudate nucleus, the putamen, the globus pallidus externus, and the globus pallidus internus. The caudate nucleus and putamen are sometimes referred to as a corpus striatum, while the putamen, globus pallidus externus, and internus are sometimes referred to as a lentiform nucleus. The basal ganglia are closely related to the substantia nigra of the midbrain and the subthalamic nuclei of the diencephalon. Information in the basal ganglia passes through two different pathways. The direct pathway leads to the stimulation of the motor cortex, while the indirect pathway leads to inhibition of the motor cortex.
The human brain is one of the most important and sophisticated organs of the human body. In fact, every minute it receives about 15% of the total blood pumped by the heart to our entire body. Cerebral circulation is a complex circulatory system formed by the two internal carotid arteries, the two vertebral arteries, and their branches. The terminal branches of both the internal carotid arteries and the vertebral arteries lie in the subarachnoid space, which is a space between two meningeal layers, called the arachnoid mater and pia mater. Anastomoses between these two arteries and their branches give rise to the circle of Willis, which is a system of vessels at the base of the brain that helps to ensure adequate blood flow to this vital organ. Let's start off with the internal carotid arteries, or ICAs, which are the terminal branches of the common carotid arteries, and form the anterior part of the cerebral vascular system. The ICAs ascend on both sides of the neck to reach the base of the skull, where they enter a passageway in the petrous part of the temporal bone, called the carotid canal. Within the carotid canal, the ICA is close to venous plexuses as well as the carotid plexuses of sympathetic nerves. Within the canal, each ICA turns 90 degrees anteromedially, then another 90 degrees superiorly to exit the carotid canal and enter the cranial cavity. Inside the cranial cavity, the ICA runs through the cavernous sinus, which is in fact a dural venous sinus. So the ICA, an artery, actually runs through a sinus filled with venous blood. Within the cavernous sinus, the ICA travels alongside the abducens nerve and lies in proximity to the oculomotor nerve, the trochlear nerve, and the ophthalmic and maxillary divisions of the trigeminal nerve. Then the ICA emerges from the cavernous sinus and divides into the anterior cerebral artery, the middle cerebral artery, and several smaller branches. To simplify this complex course, the ICA can be divided into four parts: the cervical part, that extends from common carotid to the carotid canal, the petrous part, that extends from the carotid canal to the foramen lacerum, the cavernous part, meaning the part of the ICA within the cavernous sinus, and the cerebral part, after it exits from the cavernous sinus. The cerebral part of the ICA extends by bifurcating into two major terminal branches, the anterior cerebral artery and middle cerebral artery. Now, the cerebral part of the ICA gives off several other collateral branches that supply various structures in the head, including the brain. Anteriorly, right after it exits from the cavernous sinus, it gives off the ophthalmic artery. This artery intuitively enters the orbit and gives off the central retinal artery, which is the main blood supply to the retina. Posteriorly, the ICA gives off two branches, the posterior communicating artery and the anterior choroidal artery. The posterior communicating artery arises right before the terminal bifurcation of the ICA and connects the middle cerebral and posterior cerebral artery in the circle of Willis. The anterior choroidal artery also arises close to the terminal bifurcation and courses posteriorly to enter the lateral ventricle of the brain, to end in the choroid plexus, supplying it and several other cerebral structures along its course. And now let's use an image of the medial side of one cerebral hemisphere to look at the two terminal branches of the ICA, the anterior cerebral artery and the middle cerebral artery. From its origin, the anterior cerebral artery runs anteriorly and then turns superiorly and travels posteriorly, arching over the corpus callosum. Throughout this course, it supplies the medial aspect of the frontal lobe and the parietal lobe by giving off numerous small cortical branches. These cortical branches will also arch over the lateral aspect of the hemisphere and supply the superolateral part of the frontal and parietal lobes. The two anterior cerebral arteries on each side are connected through the anterior communicating artery, forming the anterior part of the circle of Willis. Next, after branching from the internal carotid artery, the middle cerebral artery travels laterally along the lateral sulcus. While it traverses the lateral aspect, it gives off deep penetrating branches called central arteries, which supply the deeper structures of the brain, like the corpus striatum, which is made up of the caudate and lenticular nucleus. This is why these central arteries are often referred to as lenticulostriate branches of the middle cerebral artery. After reaching the lateral surface, the middle cerebral artery provides a few branches that supply the lateral surface of the temporal lobe and part of the infralateral surface of the frontal and parietal lobe. Now, both the anterior and middle cerebral arteries are often referred to as terminal arteries, as there is no collateral circulation in areas supplied by them. Thus, any blockage in these arteries may prevent blood from reaching that part of the brain completely. Okay, that was a lot. Let's take a short break and see if you can recall the collateral and terminal branches of the internal carotid artery. All right, now let's switch gears and look at the vertebral arteries, which arise from the subclavian arteries and form the vertebro-basilar system. The vertebral arteries ascend through the transverse foramina of the cervical vertebrae to enter the skull through the foramen magnum, and then they pierce the dura mater to get into the subarachnoid space. Here, the vertebral arteries start their intracranial course by moving forward, upward, and medially over the medulla oblongata. At the pontomedullary junction, which is where the pons and the medulla meet, the two vertebral arteries merge with each other and form a single basilar artery, hence the term vertebro-basilar system. Now, before the two vertebral arteries merge, they give off two important branches, the anterior spinal artery and the posterior inferior cerebellar artery. The anterior spinal artery is formed by two separate branches of the vertebral artery, which travel inferiorly and medially, and subsequently form the singular anterior spinal artery at the level of the foramen magnum. The anterior spinal artery then courses in the anterior median aspect of the spinal cord, supplying its rostral anterior 2/3. The posterior inferior cerebellar artery, or pica, on the other hand, originates from the vertebral artery and winds posteriorly around the upper part of the medulla oblongata, travels along the inferior aspect of the cerebellum, and divides into medial and lateral branches. The medial branch continues posteriorly, while the lateral branch supplies the inferior part of the cerebellum. Now, there is another branch called the posterior spinal artery, which in 1/4 of the population arises from the vertebral artery, and in the other 3/4 of the population from the pica. The posterior spinal artery mainly supplies the posterior aspect of the spinal cord. Now let's look at the basilar artery. After its origin, it ascends along the ventral surface of the pons. On its way, it gives off 4 paired collateral branches: the anterior inferior cerebellar arteries, or ICA, the labyrinthine arteries, the pontine arteries, and the superior cerebellar arteries. Then, just superior to the origin of the oculomotor nerve, the basilar artery ultimately divides into a pair of terminal branches, called the posterior cerebral arteries. The left and right posterior communicating arteries originate from these posterior cerebral arteries and merge with the internal carotid arteries, thereby closing the posterior portion of the circle of Willis. The anterior inferior cerebellar arteries course posterolaterally and mainly supply the inferior aspect of the cerebellum, as well as part of the pons and middle cerebellar peduncle. Next, the labyrinthine arteries course laterally, along with the 7th and 8th cranial nerves, to the internal acoustic meatus, to enter and supply the inner ear. Next up, the pontine arteries are numerous small branches that course laterally to supply the pons. Last, just before the bifurcation of the basilar artery, there are the superior cerebellar arteries, which course laterally and then travel around the cerebral peduncle to reach and supply the superior aspect of the cerebellum, the pineal gland, part of the pons, and part of the third ventricle. Okay, that was a lot. Let's take a short break and see if you can recall the branches of the vertebral artery. Great! Now let's put all this information together and look at the circle of Willis, named after the English physician Thomas Willis. See, the anterior communicating artery connects the two anterior cerebral arteries, while the posterior communicating arteries connect the posterior cerebral arteries with the internal carotid arteries on each side. This forms the circle of Willis in the subarachnoid space at the base of the brain, right in front of the midbrain. This circle helps create a collateral arterial circulation, so complete or partial blockage of any one of the cerebral arteries can be compensated by other arteries. In this way, cerebral perfusion is more likely to be maintained, which helps to prevent ischemic and hypoxic brain injury from occurring. The circle of Willis is closely related to some important surrounding structures. For example, the anterior communicating artery travels just anterior to the optic chiasm, while the posterior communicating artery courses just medial to the third cranial nerve on each side. Thus, any abnormal dilatation or aneurysm in a particular artery may lead to compression of a related nerve or other associated structure, causing specific neurological deficits. On a quick side note, even though the major cerebral arteries participate in the formation of the circle of Willis, each of them predominantly supplies one part of the cerebral cortex. The anterior cerebral arteries supply the anteromedial surface of the cerebral cortex. The middle cerebral arteries are located laterally, supplying the majority of the lateral surface of the cerebral cortex. Finally, the posterior cerebral arteries supply both the posterior and inferior surfaces of the cerebral cortex. An important concept to remember when considering vasculature supply in infarcts is the cortical homunculus. The homunculus is a kind of neurological map that depicts the neurons in a particular area of the cortex that process a particular body region, as well as the density of neurons that are dedicated to that region of the body. The motor homunculus is located in the frontal lobe, while the sensory homunculus is just caudal to it, in the parietal lobe. The cortical homunculus can be represented as a body lying on top of the brain, where each body part lies on top of its corresponding brain area. The toes are represented first on the medial aspect of the cerebral hemisphere. Then, moving superiorly and then laterally, more superior parts of the body are progressively represented, until we see the face most lateral. The motor and sensory homunculi on each side of the brain correspond to motor and sensory functions on the opposite side of the body. the medial area of the homunculus, representing the lower body, is supplied by the anterior cerebral artery, while the lateral area, representing the upper body and face, is supplied by the middle cerebral artery. Now, as we move away from the origin of the anterior, middle, and posterior cerebral arteries, and more towards the areas of cerebral cortex that border the cortical branches of these arteries, blood supply by these cerebral arteries gradually decreases. As a result, these border areas are more prone to ischemic injuries. In these cases, the pattern of injury is called a watershed infarct or watershed stroke, where healthy tissue continues to extract what it needs from the blood flowing by, leaving little or no oxygen and nutrients for the tissue furthest away. And as a result, the tissues that are the furthest downstream are affected the most. The furthest downstream tissues are the watershed areas, which are at the border of the blood supply from two separate groups of cerebral arteries. There are three main watershed areas where watershed infarcts typically occur: between the anterior cerebral and the middle cerebral arteries, between the posterior cerebral and the middle cerebral arteries, and between the superficial and deep branches of the middle cerebral arteries. Finally, let's briefly look at the venous drainage of the brain, which starts as a series of small superficial veins. These veins drain into dural venous sinuses, which are venous channels located between the two layers of dura mater, the periosteal layer and the meningeal layer. The superior sagittal sinus runs along the longitudinal fissure, where it absorbs cerebrospinal fluid from the meninges. It drains to the confluence of sinuses, along with the straight sinus and occipital sinuses, which then empties into the transverse sinuses. The transverse sinuses drain to the sigmoid sinuses, which then drain to the internal jugular veins, ultimately returning to the heart. All right, as a quick recap, the anterior part of the cerebral circulatory system is created by the internal carotid arteries. The ICA has four parts: the cervical part, the petrous part, the cavernous part, and the cerebral part. Collateral branches of the internal carotid artery include the ophthalmic artery, the posterior communicating artery, and the anterior choroidal artery. The terminal branches of the ICA are the anterior and middle cerebral arteries. The anterior cerebral arteries are connected via the anterior communicating artery. The posterior aspect of the cerebral circulatory system arises from the vertebral arteries, which merge to form the basilar artery. Before they join, they give off the anterior spinal artery and the posterior inferior cerebellar artery. The basilar artery gives off four paired collateral branches: the anterior inferior cerebellar arteries, or AICA, the labyrinthine arteries, the pontine arteries, and the superior cerebellar arteries. Ultimately, the basilar artery divides into a pair of terminal branches, called the posterior cerebral arteries, which in turn give off the left and right posterior communicating arteries. The circle of Willis is formed by the anterior cerebral arteries that are connected by the anterior communicating artery, and the posterior cerebral arteries and the internal carotid arteries, which are connected by the posterior communicating arteries. The three main watershed areas of the brain are between the anterior cerebral and the middle cerebral arteries, between the posterior cerebral and the middle cerebral arteries, and between the superficial and deep branches of the middle cerebral arteries. Blood returns from the brain to the systemic circulation through a series of venous channels called dural venous sinuses that ultimately drain into the internal jugular veins.
Our central nervous system is made up of the cerebrum, the cerebellum, the brainstem, and the spinal cord. The brainstem is a trunk-like part that sits in the posterior cranial fossa and connects the spinal cord inferiorly with the forebrain superiorly. The brainstem can be divided into three parts. From caudal to rostral, these parts are the medulla oblongata, the pons, and the midbrain. The brainstem is made up of white and gray matter. The white matter contains many ascending and descending fibers that act like a highway, allowing information to travel to and from the spinal cord and the higher parts of the central nervous system. Scattered amongst the white matter tracts, there are islands of gray matter that consist of neuronal cell bodies, many that are the nuclei associated with cranial nerves. Some of these collections of nuclei serve as centers for life-sustaining reflexes, like those involved with breathing and heartbeat. Others coordinate states of alertness or arousal, while others mediate motor activities and relay sensory information. First, let's look at the medulla oblongata, specifically its ventral aspect. Right in the middle, there is the anterior median fissure. On either side of it, there are two bumps called the pyramids, which contain axons of the corticospinal or pyramidal tract. Before entering the spinal cord, these fibers cross over to the opposite side, forming the decussation of the pyramids. Lateral to each pyramid, there are two oval bumps called the olives. They contain the inferior olivary nuclei, which have rich connections to the cerebellum and are involved in motor coordination and learning. Now, on the dorsal aspect of the medulla oblongata, it's visible that the rostral medulla contains the inferior part of the fourth ventricle, a space filled with cerebrospinal fluid, or CSF. Across the floor of the fourth ventricle, spreading transversely, there are the striae medullaris, which are raised stripes that contain the arcuatocerebellar fibers. Laterally, there are the inferior cerebellar peduncles, which contain fibers that travel between the medulla and the cerebellum. The fourth ventricle has a diamond or rhomboid shape, so it's also called the rhomboid fossa. The ventricle, or fossa, tapers at its caudal aspect to a point called the obex, and near this caudal limit is the entrance to the central canal of the spinal cord. In this region, CSF can travel from the fourth ventricle into the central canal. Looking at the caudal medulla, right on the midline, there's the posterior median sulcus. Next to it, there are two bumps called the gracile tubercles, which contain the gracile nuclei, one on each side. Lateral to the gracile tubercles, there are two more bumps on each side called the cuneate tubercles, which contain the cuneate nuclei. The gracile and cuneate nuclei convey fine touch, pressure, conscious proprioception, and vibratory sensations. Now let's switch gears and have a look at the embryological development of the medulla oblongata and the spinal cord. The development is important because it provides a location plan for motor and sensory nuclei and a blueprint for the overall pattern of motor and sensory information flow. If we look at a transverse section of the part of the neural tube that will develop into the spinal cord, we can see the cavity of the neural tube in the center. The neural tube has a dorsal or posterior part that forms the alar plate and a ventral or anterior part that forms the basal plate. Inside the neural tube, a groove called the sulcus limitans separates these plates. Now the alar plate gives rise to sensory nuclei and later develops into the dorsal horns of the spinal cord's gray matter. The basal plates, on the other hand, give rise to the motor nuclei and later develop into the ventral horns of the spinal cord. As we continue rostral along the neural tube to the part that will develop into the medulla oblongata, we can see that the cavity of the neural tube extends laterally to form the fourth ventricle. In doing so, it pushes the alar plates ventrally as well. Even though the basal and alar plates were ventrodorsal in the spinal cord, they shift to mediolateral position in the medulla oblongata. Medially, the basal plate develops into the motor nuclei of the cranial nerves, while laterally and posteriorly, the alar plate will develop into sensory nuclei. The sulcus limitans that separates them can be seen on the floor of the fourth ventricle, parallel to the posterior median sulcus. Let's take a quick break and see if you can identify structures of the dorsal aspect of the medulla oblongata. Okay, now let's look at the internal structures of the medulla oblongata. And the easiest way to do this is by making a few transverse sections, starting with the first one at the level of the caudal medulla. On the ventral aspect, there are two pyramids which contain descending fibers of the corticospinal tracts. The majority of these fibers cross over to the opposite side of the pyramidal decussation and form the lateral corticospinal tract as the fibers enter the spinal cord. Lateral to the pyramids, there is the anterior spinocerebellar tract, and behind it, the posterior spinocerebellar tract. These carry a proprioceptive input from the muscle spindles, Golgi tendon organs, and joint receptors to the cerebellum. Medial to them, there's the lateral spinothalamic tract, which carries somatosensory information about pain and temperature originating from below the face to the thalamus. Between these tracts and the decussation of the pyramids, part of the inferior olivary nucleus can be seen. On the dorsal aspect, lateral to the posterior median sulcus, the rostral part of the fasciculus gracilis can be seen on both sides, and just lateral to those fibers, the fasciculus cuneatus. Ventral to them, there are the nucleus gracilis and nucleus cuneatus, which are made up of gray matter. Lateral to the fasciculus cuneatus, the spinal trigeminal tract is present, and ventral to it, the spinal trigeminal nucleus. Finally, between all these nuclei and tracts, there's the reticular formation, which is a network of nuclei with interspersed afferent and efferent fibers. It stretches across the entire brainstem, so it acts like an interface between the spinal cord and higher brain centers. The reticular formation influences many functions, such as motor reflexes, eye movements, autonomic functions, and even consciousness. Now let's look at another transverse section, this time at the level of the decussation of the medial lemnisci. Here, there's a somatosensory tract decussation. Axons originating from the nucleus gracilis and nucleus cuneatus form the internal arcuate fibers that pass around the central gray matter. Ventral to it, these fibers cross the neural axis to the opposite side. Dorsal to the decussation in the central gray matter, there are the hypoglossal nuclei. Other structures are similar to those described in the previous section. Now, even more rostrally, let's look at the olives and inferior cerebellar peduncles. Here, instead of the central canal dorsally, there is a space created that makes up the fourth ventricle. The central gray matter is larger and sits under the floor of the fourth ventricle, where it contains nuclei of cranial nerves next to the posterior median sulcus. From medial to lateral, these are the hypoglossal nuclei, dorsal motor nuclei of vagus, and the solitary nucleus. Dorsal to the solitary tract, there is the medial vestibular nucleus, and laterally, the inferior vestibular nucleus. Ventral to all these nuclei, there's the reticular formation, and buried deep within it, the nucleus ambiguus. Lateral to the reticular formation, there are the spinal trigeminal tracts and nuclei, and most laterally, the inferior cerebellar peduncles. On the ventral side, next to the anterior median fissure, there are the pyramids. They contain the corticospinal or pyramidal fibers that descend to the spinal cord, and the corticobulbar fibers that target motor nuclei of the cranial nerves in the brainstem. Dorsal and lateral to the pyramids, there are the inferior olivary nuclei that look like two big crumpled bags, with their openings positioned toward the middle. These nuclei send fibers across the midline toward the inferior cerebellar peduncles. Also dorsal to the pyramids, and between the inferior olivary nuclei, there are the medial lemnisci, whose axons originated from the nucleus gracilis and nucleus cuneatus in the medulla. Dorsal to the medial lemnisci, and between the reticular formation, there are the tectospinal tracts that coordinate head and eye movements. Between the tectospinal tracts and the hypoglossal nuclei, there are the medial longitudinal fasciculi, or MLF, which connect the vestibular and cochlear nuclei with the motor nuclei of the cranial nerves 3, 4, and 6. Lastly, between the reticular formation and inferior olivary nuclei are the lateral spinothalamic tracts, and laterally, near the surface of the medulla, are the anterior spinocerebellar tracts. Finally, let's look at the most rostral transverse section at the level just below the pons. Here, instead of inferior vestibular nuclei, there are the lateral vestibular nuclei, positioned between the inferior cerebellar peduncles and the reticular formation. Also, dorsal to the inferior cerebellar peduncles and lateral to the fourth ventricle are the dorsal cochlear nuclei, and just ventral to those, we find the ventral cochlear nuclei. Let's take a quick break and see if you can recall the nuclei of the medulla oblongata. Let's continue with the next part of the brainstem, which is the pons. Pons means bridge in Latin, so the pons is like a bridge between the medulla oblongata and the midbrain, ventral to the cerebellum. On its ventral aspect, there are two middle cerebellar peduncles on each side that are connected by a curved surface that looks like a bridge. In the middle of it is a shallow depression called the basilar groove, which holds the basilar artery. Switching gears, the pons also has a dorsal aspect, which is triangular shaped. It creates the rostral half of the floor of the fourth ventricle. Laterally, there are the superior cerebellar peduncles. Running vertically down the midline stretches the median sulcus, and just parallel to it on each side, the sulcus limitans. Between the median sulcus and the sulcus limitans, there are two elevations called the medial eminences, whose caudal parts enlarge to form the facial colliculi. The facial colliculi are two bumps, which are made up of axons from the facial nuclei that wrap around the abducens nuclei. Laterally, from the sulci limitans, are the vestibular areas that contain the vestibular nuclei. Caudal to the superior cerebellar peduncles, we see the middle cerebellar peduncles. Now let's make a transverse section through the pons at the level of the facial colliculi. Dorsally, there's the cavity of the fourth ventricle, and ventral to it, the reticular formation. Ventral to the facial colliculi are the abducens nuclei, and ventrolateral to them are the facial nuclei. The axons of the facial nuclei travel posteromedially to wrap around the abducens nuclei and then continue ventrolaterally. Lateral to the facial nuclei, we find the spinal trigeminal nucleus, and dorsal to them and lateral to the abducens nuclei are the medial vestibular nuclei. The motor tracts on this section are the corticospinal and corticobulbar tracts on the ventral side and the MLF found between the facial colliculi around the median sulcus. As for the sensory tracts, there are the medial lemnisci, ventral to the reticular formation, spinal thalamic tracts, lateral to the reticular formation, lateral lemnisci, which carry auditory information, positioned lateral to the medial lemnisci, and the spinal trigeminal tracts next to the spinal trigeminal nuclei. Let's look at another transverse section, now at the level of the trigeminal nuclei. Here we can see the motor trigeminal nuclei that sit ventral near the ventrolateral corner of the fourth ventricle, and the main sensory trigeminal nuclei lateral to them. Other structures are similar to the previous section. Let's take a break and see if you can identify the nuclei of the pons, as well as the important motor and sensory tracts. Finally, let's look at the midbrain, which sits between the pons and the forebrain. First, let's look at its ventral aspect. Laterally, there's the crus cerebri, or the basis pedunculi, which contain descending motor tracts from the cerebrum. Between them, there's a space called the interpeduncular fossa. The gray matter at the junction of the crus cerebri at the base of this fossa is called the posterior perforated substance because many small blood vessels penetrate its surface. Switching to the dorsal aspect, we can see four bumps, or colliculi, called the corpora quadrigemina. The superior colliculi contain nuclei involved in visual reflexes, while the inferior colliculi contain nuclei that are part of the auditory pathway. Lateral to the colliculi, there are two elevations of brachia, ascending into the antero-posterior direction. The superior brachium, or brachium of the superior colliculus, stretches from the lateral geniculate body and the optic tract to the superior colliculus. It contains afferent fibers carrying information from the retina, occipital lobe, and frontal eye fields of the frontal lobe. The inferior brachium, or the brachium of the inferior colliculus, stretches from the inferior colliculus to the medial geniculate body, carrying auditory information. let's make a transverse section through the midbrain at the level of the inferior colliculi. First, regarding its general organization, the midbrain can be divided into three main parts: the tectum, tegmentum, and crus cerebri. The tectum contains both the superior and inferior colliculi and is located dorsal to the cerebral aqueduct, while the tegmentum lies ventrally. From the ventrolateral side of the tegmentum, the crus cerebri expand and are separated medially by the interpeduncular fossa. The crus cerebri, together with the respective half of the tegmentum, are referred to collectively as the cerebral peduncle. Around the cerebral aqueduct lies the central gray matter, or the periaqueductal gray matter. Now let's inspect the internal structures of the midbrain. First up, the nuclei, which are the nuclei of the inferior colliculi, found dorsally behind the central gray matter. The mesencephalic trigeminal nuclei, sitting in the central gray matter lateral to the cerebral aqueduct, the trochlear nuclei in the ventral part of the central gray matter near the middle, and the substantia nigra, meaning black substance, found between the tegmentum and crus cerebri, separating them. The cell bodies found in the substantia nigra have granules filled with a pigment called neuromelanin, giving them a darker appearance. The substantia nigra is part of the basal ganglia, which is a system of central nervous system components that help integrate and regulate various types of information, primarily motor information. In the tegmentum, between the central gray matter and the substantia nigra, we find the reticular formation again. Moving to the motor tracts, these are the MLF, anterior to the trochlear nucleus, and the corticospinal and corticobulbar tracts, found within the middle third of the crus cerebri. Lastly, the sensory tracts are positioned on the lateral sides of the tegmentum, behind the substantia nigra. Starting ventromedially, these are the medial lemnisci, the spinal thalamic tracts, the trigeminal lemnisci, and the lateral lemnisci. Finally, let's move rostrally again to the level of the superior colliculi. The nuclei of the superior colliculi are posterior to the central gray matter. Lateral to them, we find the pretactile nuclei that receive input from the retina about the presence of light. They send signals to the parasympathetic nuclei of the oculomotor nerve, also known as the Edinger-Westphal nuclei, situated in the ventral portion of the central gray matter. These two nuclei are involved in the pupillary light reflex. In the ventral region of the central gray matter, near the middle, are the oculomotor nuclei as well. Ventrolateral to them, in the tegmentum, there are two large nuclei called the red nuclei, which influence the tone of contraction for flexor musculature. Lateral and posterior to the red nucleus is the reticular formation. The mesencephalic trigeminal, substantia nigra, and motor and sensory tracts are present like in the previous section, but here the lateral lemniscus is missing as it has already terminated on the nuclei of the inferior colliculus. Before the recap, let's see if you can identify the nuclei of the midbrain as well as the important motor and sensory tracts. All right, as a quick recap, the brainstem is made up of the medulla oblongata, the pons, and the midbrain. On the ventral aspect of the medulla, there are the pyramids and the olives. Dorsally, there are the inferior cerebellar peduncles, the floor of the fourth ventricle, stria medullaris, and the entrance to the central canal and the gracile and cuneate tubercles. At the level of the decussation of the pyramids, we can find the central canal, pyramids, decussation of the pyramids, nuclei, and fasciculi gracilis and cuneatus, spinal tracts and spinal nuclei of trigeminal, inferior olivary nuclei, ventral and dorsal spinocerebellar tracts, and the lateral spinothalamic tracts. At the level of the decussation of the medial lemnisci, there are the hypoglossal nuclei. At the level of the olives, there are the fourth ventricle, inferior olivary nuclei, vestibular nuclei, and hypoglossal nuclei, as well as the nucleus ambiguus, dorsal motor nucleus of vagus, solitary nucleus, MLF, medial lemnisci, and tectospinal tracts. Just caudal to the pons, we can see the lateral vestibular nuclei and the cochlear nuclei. On the ventral aspect of the pons, we see the ventral parts of the middle cerebellar peduncles and the basilar groove. On the dorsal aspect are the superior and middle cerebellar peduncles, median sulcus, medial eminence and facial colliculus, sulcus limitans, and vestibular area. At the level of the facial colliculi, there are the facial, abducens, and medial vestibular nuclei, and the trigeminal lemnisci, and the motor and main sensory nuclei of trigeminal, corticospinal, and corticobulbar tracts, the MLF, spinothalamic tracts, and medial lemnisci. The midbrain contains the crus cerebri and the interpeduncular fossa on the ventral aspect and the corpora quadrigemina on the dorsal aspect, along with the superior and inferior brachii and medial and lateral geniculate bodies on the lateral side. At the level of the inferior colliculi, there are nuclei of the inferior colliculus, trochlear nuclei, substantia nigra, corticospinal and corticobulbar tracts, MLF, medial lemnisci, lateral lemnisci, trigeminal lemnisci, and the spinothalamic tracts. At the level of the superior colliculi, there are the nuclei of superior colliculus, ocular motor nuclei, pretectal nuclei, Edinger-Westphal nuclei, and red nuclei.
The word cerebellum translates to little brain. Not because it's the brain of a tiny animal or baby, but rather because the fact that the cerebellum looks like a smaller version of the human cerebrum. Very simply, the cerebellum assists with coordinating and adjusting voluntary movement. It plays a major role in posture, balance, maintenance of muscle tone, and coordinating skilled voluntary motor activities, things like riding a bicycle, or, for the more adventurous, walking a tightrope. In order for the cerebellum to undertake these functions, it has to be in constant communication with the cerebral cortex. It also sends and receives signals to many other structures in the central and peripheral nervous systems, processing information about current movement and positional states in order to help refine, correct, and improve the motion. Now, the cerebellum sits in the posterior part of the cranium, called the posterior cranial fossa, and it's covered by the tentorium cerebelli, which separates the cerebellum from the occipital and temporal lobes of the brain. Anterior to the cerebellum lies the fourth ventricle, pons, and medulla oblongata. Just like the cerebrum, the cerebellum consists of two hemispheres. These two hemispheres are connected by a narrow ridge in the middle called the vermis. From an inferior view, parallel to the vermis, there are two distinguishable lobules called the cerebellar tonsils. The cerebellum can be divided into three lobes: the anterior lobe, the posterior lobe, and the flocculonodular lobe. From a superior view, we can identify the anterior lobe, functionally referred to as the spinocerebellum, which is responsible for the regulation of muscle tone and adjusting ongoing movements. Posterior to the anterior lobe is the V-shaped primary fissure. From a superior view, and posterior to this primary fissure, is the posterior lobe, functionally referred to as the cerebrocerebellum, or pontocerebellum, which contains a horizontal fissure, separating the superior and inferior surface of the cerebellum. The cerebrocerebellum is the largest part of the cerebellum, and is responsible for assisting in planning and programming of skilled or fine motor movements. Looking at the cerebellum from an anterior view, the posterior lobe is bounded by the posterolateral fissure. This fissure separates the posterior lobe from the third lobe of the cerebellum, called the flocculonodular lobe, or functionally referred to as the vestibulocerebellum. The flocculonodular lobe is responsible for the maintenance of posture and balance. The flocculonodular lobe is named such because it contains a central part of the vermis called the nodule, as well as two lateral flocculi. If we continue to view the cerebellum from the anterior aspect, we can see bundles of dense white matter that attach the cerebellum to the brainstem. These white matter stalks are called cerebellar peduncles and consist of superior, middle, and inferior divisions. They contain efferent and afferent axons that signal back and forth between the cerebellum and the central nervous system. The superior cerebellar peduncle connects the cerebellum with the midbrain. The middle cerebellar peduncle connects with the pons, and the inferior cerebellar peduncle attaches to the medulla oblongata. Afferent fibers to the cerebellum can be found within all three cerebellar peduncles, but the majority of afferent signals use the inferior and middle peduncles for passage. Efferent signals from the cerebellum, however, travel mainly through the superior peduncle. On a sagittal section, the cerebellum looks similar to the cerebrum, in that the cortex is folded, creating ridges with small sulci in between. The difference, however, is that in the cerebellum, the cortical ridges are thinner, smaller, and organized into more parallel layers, which are called folia. These folia not only increase the surface area, but enable the large area of cortex to fit into a smaller space, just like the cerebrum. The folia contain an external gray matter layer, called the cerebellar cortex, and a subcortical white matter region deep to the external gray matter. As we see, the shape of this white matter within the folia creates a tree-like arrangement, or branching pattern, referred to as an arbor vitae, or tree of life. On a transverse section of the cerebellum, we can see 4 clusters of deep gray matter nuclei buried deep within the subcortical white matter. These deep cerebellar nuclei, or intracerebral nuclei, contain multipolar neurons that receive signals from the cerebellar cortex and other parts of the nervous system, and their axons contribute to the formation of the 3 cerebellar peduncles. From lateral to medial, these deep cerebellar nuclei consist of the dentate, emboliform, globose, and fastigial nuclei. To remember these, remind yourself that in order to have a healthy cerebellum, you don't eat greasy foods. In addition to having anatomical divisions, the cerebellar cortex can also be divided into 3 functional regions that are positioned longitudinally. The most lateral and largest functional region is the lateral zone. The lateral zone sends signals to the dentate nucleus, the largest of the deep cerebellar nuclei, and together they assist in planning and programming movements. Medial to the lateral zone is the intermediate zone, also known as the paramedian or paravermal zone. The intermediate zone sends signals to the emboliform and globose nuclei. Collectively, these 2 nuclei are known as the interposed nuclei and are usually referred to together as they both work in the intermediate zone. Finally, most medial and occupying the cortex of the vermis is the 3rd functional zone, the median or vermal zone. The median zone will send signals to the fastigial nucleus, which is the most medial of the deep cerebellar nuclei, located within the vermis and next to the roof of the 4th ventricle. The intermediate and median zones, along with their deep cerebellar nuclei, are involved in modulating motor execution of lateral and medial descending motor pathways, respectively. Let's take a quick break and see if you can identify the lobes of the cerebellum, as well as the functional zones and deep cerebellar nuclei. Now let's have a look at the afferent pathways, which bring information to the cerebellum to be processed, and the efferent pathways, which leave the cerebellum to help coordinate motor activity. Afferent pathways generally originate from the spinal cord and brainstem, the cerebral cortex, and the vestibular system. Starting with the afferent pathways from the spinal cord to the cerebellum, let's look at the ventral or anterior spinocerebellar pathway first. It carries proprioceptive information from muscle spindles, Golgi tendon organs, and joint receptors of the lower extremities. Then, the afferent fibers enter the spinal cord, where they synapse with spinal border cells located in lamina 7 of the spinal cord gray matter. From here, the majority of these axons cross to the contralateral side of the spinal cord and form the ventral spinocerebellar pathway, which ascends in the white matter of the spinal cord to the brainstem. Here, the axons cross back over and enter the cerebellum through the superior cerebellar peduncle to reach the cerebellar cortex. The signals on the ventral spinocerebellar pathway cross over the neural axis and then cross back, so it's often referred to as the double crosser. Next is the dorsal or posterior spinocerebellar pathway. This pathway contains fibers that receive proprioceptive information from muscle spindles, Golgi tendon organs, and joint receptors mainly found in the trunk and lower extremities. This information enters the spinal cord from peripheral nerves and the signal synapses on Clark's nucleus, or Clark's column, also known as nucleus dorsalis. Instead of crossing over after they synapse, the axons ascend in the ipsilateral white matter of the spinal cord to the brainstem, where they then enter the cerebellum through the inferior cerebellar peduncle to reach the cerebellar cortex. The final afferent pathway that carries proprioceptive information from the extremities is called the cuniocerebellar pathway. The axons in this pathway receive proprioceptive information from muscle spindles, Golgi tendon organs, and joint receptors within the upper limb and upper thorax. Signals within this pathway synapse in the external or accessory cuneate nucleus located in the medulla. The axons travel ipsilaterally through the inferior cerebellar peduncle to reach the cerebellar cortex. Let's now move on to the afferent pathways from the cerebral cortex to the cerebellum, which include the corticopontocerebellar, cerebro-olivocerebellar, and cerebro-reticulocerebellar pathway. Talk about your tongue twisters. Okay, now these pathways, originating in the cerebral cortex, signal through brainstem structures to reach the cerebellum and allow for these areas to communicate to further regulate and modify voluntary movements. For example, the initiation, planning, and timing of motor activities. This information is important to the cerebellum to know so that it can take part in making the appropriate adjustments and modifications to that plan for synergy and overall motor coordination. The corticopontocerebellar pathway is important in relaying motor commands of the cortex and begins with the afferent fibers arising from the cerebral cortex of the frontal, temporal, parietal, and occipital lobe. These fibers signal through the corona radiata and internal capsule to synapse on pontine nuclei. Then the signals are sent along transverse fibers, called pontocerebellar fibers, that cross over and enter the middle cerebellar peduncle to terminate in the cortex of the contralateral cerebellar hemisphere. The cerebro-olivocerebellar pathway also begins in the cerebral cortex of the four lobes and sends its fibers through the corona radiata and internal capsule, where the fibers synapse in the inferior olivary nuclei. After synapsing, these fibers cross the midline and travel through the inferior cerebellar peduncle to synapse with the contralateral cerebellar hemisphere. The cerebro-reticulocerebellar pathway also arises from the cerebellar cortex, however, mainly from the sensor-and-motor cortical regions associated with the parietal lobe. The axons descend in the same way as the previous two pathways and synapse with the nuclei of the reticular formation located in the pons and medulla. These fibers then travel through the middle and inferior cerebellar peduncles to terminate in the cerebellar hemisphere of the ipsilateral side. Finally, the last major system to provide afferent information to the cerebellum is the vestibulocerebellar pathway. It plays a key role in the maintenance of balance, posture, body position, and coordination of eye movements. This pathway begins by receiving sensory input regarding motion from the semicircular canals of the inner ear, as well as body position relative to gravity from the saccule and utricle via the vestibular nerve, a portion of the vestibulocochlear cranial nerve. During their course, afferent fibers of the vestibular nerve either signal straight through the inferior cerebellar peduncle to the ipsilateral cerebellar cortex, or they synapse first in the vestibular nuclei of the brainstem before signaling to the cerebellum. Together, these afferent fibers reach the cerebellar cortex of the flocculonodular lobe to be processed. The vestibulocerebellar pathway gathers visual input as well. These afferent fibers originate from the superior colliculus and the primary visual cortex, and the visual information is relayed through the superior cerebellar peduncle to reach the flocculonodular lobe. Okay, now that was a lot to take in. Let's take a quick quiz and see if you can identify the afferent tongue twisters, pathways, from the brain to the cerebellum on this image. So now that we've discussed the major afferent pathways of the cerebellum, what does the cerebellum do with all of the information it's received? Well, the cerebellum processes this information, and the cerebellar cortex relays a movement modification message to the deep cerebellar nuclei, which then sends signals along different afferent fibers to places such as the vestibular nuclei, thalamus, red nucleus, reticular formation, cerebral cortex, and the spinal cord. In doing so, the cerebellum plays a continuous role in maintaining posture, balance, and modifying, adjusting, and coordinating movements of the body. The first efferent pathway we're going to discuss is the vestibular pathway, which is responsible for regulating extensor muscle tone. Fibers originate in the vestigial nucleus of the cerebellum and travel through the inferior cerebellar peduncle to synapse on the lateral vestibular nuclei. From here, some efferent fibers form the vestibulospinal tracts, which signal to motor neurons of the spinal cord that control anti-gravity musculature that helps maintain posture. Other efferent fibers will form the medial longitudinal fasciculus, which transmits information to the motor nuclei of cranial nerves 3, 4, and 6 to modify and control movements of the eye. Another important efferent pathway of the cerebellum is the dentatothalamic pathway, which is responsible for modifying ipsilateral motor activity. This pathway begins in the dentate nucleus of the cerebellum, where fibers cross the midline and travel through the superior cerebellar peduncle to the contralateral ventrolateral nucleus of the thalamus. The fibers synapse in the thalamus, and from there, the signal continues through the internal capsule and corona radiata to reach the primary motor cortex. Once here, the information is relayed to motor pathways such as the corticospinal tract, and this is how the cerebellum influences and modulates motor activity of descending motor pathways. Next, there's the globose emboliform rubral pathway, which also influences ipsilateral motor activity. These efferent fibers begin in the globose and emboliform nuclei, travel across the midline through the superior cerebellar peduncle, and synapse with the contralateral red nucleus. This pathway influences motor activity of the rubrospinal tract, which acts on proximal flexor musculature of the upper limb. Finally, we have the fastigial reticular pathway, where efferent signals originate in the fastigial nucleus, travel through the inferior cerebellar peduncle, and then synapse with neurons in the reticular formation. These signals provide modulatory information to the medial and lateral reticulospinal tracts, which collectively are involved in regulation of muscle tone and posture. Alright, as a quick recap. The cerebellum is located in the posterior cranial fossa, below the occipital and temporal lobes, and ventral to it is the fourth ventricle, pons, and medulla oblongata. It has two hemispheres which are connected by the vermis and is divided into 3 lobes. The primary fissure divides the anterior and posterior lobes, while the posterolateral fissure separates the posterior lobe from the flocculonodular lobe. From an anterior view, the cerebellum is connected to the rest of the central nervous system via the superior, middle, and inferior cerebellar peduncles. Looking at a cross-section of the cerebellum, it's composed of gray matter folds with white matter within that form folia. The 4 deep gray matter nuclei are the dentate, emboliform, globose, and fastigial nuclei. The cerebellar cortex can be divided into 3 functional zones: the median, which helps to adjust medial descending motor pathways that include those that act on axial musculature, the intermediate or paramedian zone, which assists in adjusting lateral descending motor pathways that include those that act on appendicular musculature, and the lateral zone that works on planning and evaluating movements. Major afferent pathways from the spinal cord to the cerebellum include the ventral spinocerebellar pathway, the dorsal spinocerebellar pathway, and the cuneo-cerebellar pathway. Major afferent pathways from the brain to the cerebellum include the corticopontocerebellar pathway, the cerebro-olivocerebellar pathway, and the cerebro-reticulocerebellar pathway. The vestibulocerebellar pathway gathers information from the semicircular canals, inner ear, superior colliculi, and primary visual cortex to help maintain balance, posture, body position, and coordinate eye movements. And finally, the efferent pathways from the cerebellum include the vestigial vestibular pathway, the dentatothalamic pathway, the globose emboliform rubral pathway, and the vestigial reticular pathway. Now, we know what you're thinking. Don't worry, here at Osmosis, we're not telepathic. But by watching this video on the cerebral cortex, we know you have the brain on your mind. So let's get to it. The human central nervous system basically consists of the spinal cord and the brain, which includes the cerebrum, diencephalon, cerebellum, and brainstem. Taking a closer look at the cerebrum, it consists of two nearly symmetrical halves, called the cerebral hemispheres, and the basal ganglia, also referred to as basal nuclei. Furthermore, each cerebral hemisphere is divided into four main lobes: the frontal, parietal, temporal, and occipital, as well as what's become to be known as the fifth lobe, the insula, or insular cortex. If we were to cut through the cerebral hemispheres in the coronal plane, which means transecting from left to right and dividing the brain into rostral and caudal divisions, we would see the cerebral cortex. This is the outermost area of the cerebral hemispheres, and it's composed of gray matter containing billions of nuclei, or neuronal cell bodies. A cell body and its dendrites, along with its axon and synaptic terminal, collectively make up a structure called a neuron. Neurons thus allow for information processing and communication with other neurons within the nervous system. The gray matter gets its name from its dark appearance during gross inspection. Deep to the gray matter is the subcortical white matter, which is made up of myelinated axons connected to the nuclei of the gray matter. White matter gets its name because the myelination of the axons gives this area a white appearance on gross inspection. The largest white matter tract is the corpus callosum, which sends signals between the two cerebral hemispheres, essentially connecting them together. Found throughout the subcortical white matter are further collections of gray matter masses containing neuronal cell bodies referred to as the basal ganglia, also called the basal nuclei. The basal ganglia consist of the caudate and putamen, the globus pallidus, the subthalamic nucleus, and the substantia nigra. Note that the term striatum refers to the caudate and putamen. The term lentiform nuclei, or lenticular nuclei, refers to the putamen and globus pallidus, and the term corpus striatum refers to all three structures, the caudate, the putamen, and the globus pallidus. All of the structures of the basal ganglia have their own unique functions and pathways. Running in between these basal ganglia are more white matter afferent and efferent axon tracts, the most notable being the internal capsule. The internal capsule is a collection of densely packed white matter, or axons, which divides the corpus striatum and acts like a highway for information flow between the cerebral cortex and the brainstem and spinal cord. Generally, the right cerebral hemisphere sends and receives signals from the left side of the body, while the left hemisphere sends and receives signals from the right side of the body. Now, taking a step back and looking at the external surface of the brain, we can see that the cerebral cortex is not flat, but covered with many folds called gyri, which are separated by shallow grooves called sulci, or by deeper grooves or clefts called fissures. One function of these gyri and sulci is to allow nearly 2.5 square feet of the cerebral cortex to fold in on itself, allowing it to fit within the small space of the neurocranium, just like the folds of an accordion when it's closed. A second advantage of this cortical folding is that it effectively increases the surface area, allowing more nuclei to be packed into the cortex. This greater number of nuclei allows for more signaling and therefore more advanced processing and higher cortical function. The deep fissures also help to separate the brain into lobes. Even though the brain looks like it has a random configuration of ridges and clefts, the gyri, sulci, and fissures actually create a relatively constant pattern from person to person. This pattern can be used to identify certain external landmarks of the brain, which have very specific functions. We not only can define the cerebral cortex by lobes or functionality, which we'll talk about soon, but the cerebral cortex can also be divided into histologically similar regions called Brodmann's areas. This means that the neurons within a particular Brodmann's area are all arranged in the same manner and partake in a similar function. Approximately 180 Brodmann's area have been identified in the human brain through MRI and other mechanisms. We'll mention Brodmann's area for some of the primary areas of cortex shortly. So let's take a closer look at the different sulci and deep fissures that separate the brain into 5 lobes: the frontal lobe, parietal lobe, temporal lobe, occipital lobe, and insula. When seen from above, the cerebrum has a deep midline sagittal fissure called the longitudinal fissure, which divides the brain into left and right cerebral hemispheres. Deep within this fissure is the already mentioned corpus callosum. Still looking from above, around the midline of the longitudinal fissure and moving laterally, is the coronal central sulcus, also known as the fissure of Rolando, which separates the frontal lobe rostrally, or anteriorly, from the parietal lobe caudally, or posteriorly. The rostralmost point of the frontal lobe is called the frontal pole. The central sulcus can also be seen on a lateral view of the brain, traveling inferiorly across the lateral aspect of the hemispheres. From this lateral view, beneath the frontal and parietal lobes is a lateral fissure, also known as a lateral sulcus or Sylvian fissure. The lateral fissure is found mainly on the lateral and inferior surface of the cerebral hemispheres, and it separates the frontal and parietal lobes above from the temporal lobe below. The lateral fissure extends in 3 directions: rostrally as the anterior ramus, superiorly as the ascending ramus, and caudally as the posterior ramus. The most rostral point of the temporal lobe is called the temporal pole. Furthermore, the area of the cortex called the insula, or insular cortex, lies at the bottom of the lateral fissure, hidden from the external surface of the brain. So we need to open up the folds of the lateral fissure or dissect them in order to see it. The insula has the central sulcus of the insula running through it, forming both short gyri, rostral to the sulcus, and long gyri, caudal to the sulcus. Looking at the brain from a posterior view, around the posterior middle portion of the cerebral hemisphere, there's a fissure called the parieto-occipital fissure, which travels inferiorly and anteriorly. The parieto-occipital fissure separates the parietal lobe rostrally from the occipital lobe caudally. And from a medial view of the hemisphere, the parieto-occipital sulcus is joined halfway by the calcarine sulcus. The most caudal point of the occipital lobe is called the occipital pole. Now that we've outlined the general boundaries of each lobe, let's look at the components of each lobe in more detail, starting with the frontal lobe. On the lateral aspect, rostral to the central sulcus, is the precentral sulcus that runs parallel to it. Together, the central sulcus and the precentral sulcus form the borders of the precentral gyrus. From the rostral side of the precentral sulcus arise two more horizontal sulci, the superior frontal sulcus and the inferior frontal sulcus, which extend toward the frontal pole. These divide the rest of the frontal lobe into three gyri, and going from medial to lateral, these are the superior, middle, and inferior frontal gyri. The inferior frontal gyrus is divided into three parts by the branching rami of the lateral fissure. First, inferior to the anterior ramus, is the orbital part, or pars orbitalis. Then, between the anterior ramus and the ascending ramus, is a triangular part, or pars triangularis. And finally, posteriorly between the ascending ramus and the precentral sulcus is the opercular part, or pars opercularis. If we cut a sagittal section, meaning cutting the brain in half along the longitudinal fissure, and look at the frontal lobe from a medial view, we can also see another sulcus called the cingulate sulcus, as well as the anterior paracentral lobule, which forms the medial aspect of the precentral gyrus, and the posterior paracentral lobule, which forms the medial aspect of the postcentral gyrus, which we'll mention later. By understanding the different anatomical landmarks of the lobes, we can start to identify the different functionally specialized regions of the cerebral cortex. For example, the regions of our cortex that are responsible for controlling motor or sensory functions that assist with our everyday lives. When considering the functional areas of the frontal lobe, it contains the primary motor cortex, or Brodmann's area 4, that occupies the area of the precentral gyrus and extends over to the medial aspect of the hemisphere as the anterior paracentral lobule. The primary motor cortex houses neurons responsible for carrying out voluntary movements of different parts of our body, mainly to the contralateral or opposite side. Extending anteriorly from the primary motor cortex and over the posterior parts of the superior, medial, and inferior frontal gyri is a premotor cortex, or Brodmann's area 6. This premotor cortex receives input from other parts of the cerebral cortex, the thalamus, the basal ganglia, and directly communicates with the primary motor cortex. Its function is to assist the primary motor cortex plan and carry out voluntary movements, and therefore it's called an association cortex. The premotor cortex essentially stores and processes information about past activity and helps to integrate sensory and motor information for planning of future voluntary movements. Rostral to the premotor cortex and extending into the middle frontal gyrus is a frontal eye field, or Brodmann's area 8, which controls voluntary eye movement and allows us to move our eyes together in the same direction at the same time, known as conjugate gaze. The next area we're going to look at is Broca's area, Brodmann's area 44 and 45, which is formed by two regions of the inferior frontal gyrus, namely the pars opercularis and the pars triangularis. Broca's area is usually located in the dominant hemisphere, which in most individuals is the left hemisphere. This area has connections to the nearby motor cortex, specifically to the areas that control the muscles of the larynx, mouth, soft palate, and the tongue, as well as respiratory muscles to assist in the formation or production of words and speech. And lastly, we have the prefrontal cortex, which is located anterior to the premotor cortex and overlies the anterior portions of the superior, middle, and inferior frontal gyri. The prefrontal cortex has rich connections to other parts of the brain, and it's responsible for mainly what has been coined as executive functions, which include reasoning, planning, social behavior, judgment, and much more. Let's take our attention back specifically to the primary motor cortex, which again is responsible for controlling voluntary movements of our body. The nuclei of neurons that will control a certain region of the body are organized together, so that all the nuclei that will control the muscles of the face are organized in one specific region of cortex, while all the nuclei associated with controlling the foot are organized in another. This unique and elegant arrangement of body parts in the cortex is called somatotopy. This can be visually represented by drawing the body part above the cortical area that controls it, So it looks like this, where each body part is noted on top of its corresponding cortical area. Now, the proportion of the primary motor cortex, or the number of neurons dedicated to a particular movement, depends on how much that muscle or group of muscles is actually used. So the more a muscle is used, the more nuclei will be dedicated to it within the cortex. The number of nuclei is independent upon the size or mass of the muscle performing that movement, but it is based on how important that muscle is to your everyday life and functioning. So when we quantify the proportions of neurons in the primary motor cortex utilized by the muscles in each individual body part, we get a strange-looking humanoid figure that's called the motor homunculus, which in Latin means little human. As you can see, this primary motor cortex map creates an awkwardly large representation of the hands, fingers, and face. This means that more cortex, and hence more neurons, are dedicated to those regions, since we use them more often in our daily lives to explore our world. Going back to our primary motor cortex map, we can see that the location of the body parts is essentially inverted, anatomically, on the primary motor cortex. Starting at the superior medial part of the primary motor cortex in the anterior paracentral lobule, the toes are represented first, and then as one moves laterally through the primary motor cortex, the more superior parts of the body are progressively represented, with the more lateral and inferior part of the primary motor cortex representing the most superior parts of the body, such as the head and neck. Okay, let's move on to the parietal lobe. On the lateral view, just caudal to the central sulcus, running parallel to it, is the postcentral sulcus. Together, the central sulcus and the postcentral sulcus form the borders of the postcentral gyrus. Running caudally from the middle of the postcentral sulcus, or near it, is the intraparietal sulcus, which divides the rest of the parietal lobe into the superior parietal lobule above it and the inferior parietal lobule below it. Remember the posterior ramus of the lateral fissure? Well, there's a part of the inferior parietal lobule that folds over the end of the posterior ramus, and it's called the supramarginal gyrus. And behind it is the angular gyrus that folds over the end of the superior temporal sulcus found in the temporal lobe. This takes us to the functional regions of the parietal lobe. Similar to the primary motor cortex, there's also a primary somatosensory cortex, or Brodmann's area 3, 1, and 2, which is located in the postcentral gyrus and extends medially to the posterior paracentral lobule. The primary somatosensory cortex receives sensory input from the opposite side of the body through the ventral posteromedial, or VPM, and ventral posterolateral, or VPL, nuclei of the thalamus, enabling us to process and interpret sensory information from our body, like touch or pain. And similar to the motor homunculus, we have a sensory homunculus to visually represent the proportion of sensory fibers that the primary somatosensory cortex receives from a particular part of the body. The more sensitive a body part is, the more neurons it requires for processing sensory stimuli, and therefore it occupies a larger area of the somatosensory cortex. And again, similar to the primary motor cortex, starting from the deeper, more medial aspect of the primary somatosensory cortex in the posterior paracentral lobule, the first body part represented here is the anogenital area, followed by the foot, leg, and thigh. As we cross over to the lateral side and move inferiorly along the postcentral gyrus, the thigh is followed by the trunk, upper limb, and the head and neck, and then the intra-abdominal organs. Then, located over the superior parietal lobule, is the somatosensory association cortex, which has many connections with other sensory regions of the cerebral cortex. The somatosensory association cortex is believed to allow the ability to integrate different sensory modalities, such as being able to recognize objects through touch without visual input, like reading Braille, by comparing and associating the sensations to past sensory experiences. That was a lot of brain to take in for the brain. Let's take a moment to pause and see if you can identify some of the different regions of the brain. Now, can you label these functional areas of the brain? Now let's continue our journey and examine the temporal lobe, which lies inferior to the lateral fissure and extends posteriorly to the occipital lobe. The lateral surface of the temporal lobe includes the superior temporal sulcus and the middle temporal sulcus, which divide the temporal lobe into the superior, middle, and inferior temporal gyri. Deep inside the lateral fissure, there's also the transverse temporal gyri of Heschel, which is found on the deep upper surface of the superior temporal gyrus. Now let's look at the functional regions of the temporal lobe. Located in the transverse temporal gyri of Heschel is the primary auditory cortex, Brodmann's area 41 and 42, which receives auditory input from the medial geniculate body of the thalamus and interprets auditory information, or sound, such as when the next-door neighbors are being way too loud. Then on the dominant hemisphere, which again is most often the left, we have Wernicke's area, or Brodmann's area 22, 39, and 40, which encompasses a part of the superior temporal gyrus along with the supramarginal and angular gyri of the parietal lobe. Wernicke's area is responsible for the processing and understanding of both written and spoken language, allowing us to read a sentence, understand it, and say it out loud comprehensively. It's also connected to Broca's area by a bundle of axons called the arcuate fasciculus, which allows us to tie together speech comprehension from Wernicke's area and speech production from Broca's area. A second important function of the temporal lobe, and specifically the medial aspect of the temporal lobe, is that it houses limbic system structures related to learning, memory, and emotion, which we'll get to later. We also spoke previously about the insula, known as the fifth lobe of the brain, which is found deep in the lateral fissure. The insula is usually divided into anterior and posterior aspects. Functions are diverse and complex, with convergence of inputs from temporal, parietal, and frontal lobes. The insula has been associated with processing viscero-autonomic sensations, limbic and emotional elements, somatosensation, and possibly motor elements too. Moving on, let's get to the final lobe on our journey, the occipital lobe, which lies caudal to the parieto-occipital sulcus. The occipital lobe contains the cuneus, a wedge-shaped area bounded by the parieto-occipital fissure rostrally and by the calcarine sulcus inferiorly. Inferior to the cuneus and the calcarine sulcus is the lingual gyrus. When considering the functionality of the occipital lobe, this area of cortex is dedicated to vision, and hence a substantial part is occupied by the primary visual cortex, Brodmann's area 17. The primary visual cortex is located on the medial aspect of the hemispheres, and lines both the superior and inferior banks of the calcarine sulcus. It also extends around the occipital pole onto the lateral surface of each hemisphere. This area receives, processes, and interprets visual input from the lateral geniculate body of the thalamus. Finally, on the medial side of each hemisphere, there's a region of cortex surrounding the corpus callosum that's part of the limbic system. This region contains the subcallosal gyrus, located below the rostral part of the corpus callosum, as well as the cingulate gyrus, which begins beneath the rostral end of the corpus callosum and continues superiorly and caudally until it reaches the posterior end of the corpus callosum. The parahippocampal gyrus, which is part of the medial temporal lobe, lies rostral to the lingual gyrus of the occipital lobe and ends rostrally as the uncus, also a component of the limbic system. These cortical structures, along with the olfactory cortex, the amygdala, the hypothalamus, and the hippocampus, constitute the major structures of the limbic system. They're responsible for functions related to the preservation of the species, such as fight or flight responses, emotion, memory, and reproductive, endocrine, and other behavioral responses. Alright, as a quick recap. The cerebrum consists of two cerebral hemispheres, where the cerebral cortex is the outermost layer and consists of gray matter, which contains neuronal cell bodies or nuclei. Located beneath the gray matter is the subcortical white matter, which is composed of myelinated axons. Found within the subcortical white matter are further gray matter masses, such as the basal ganglia or basal nuclei. The external surface of the cerebral cortex is formed by gyri, sulci, and fissures. The longitudinal fissure separates the two cerebral hemispheres, which are connected via the corpus callosum. The central sulcus, parieto-occipital fissure, and the lateral fissure divide each cerebral hemisphere into four main lobes: frontal, parietal, occipital, and temporal lobes. The cerebral cortex can also be divided into histologically similar regions called Brodmann's areas, which provides a map of the primary cortices and functional regions. The main anatomical regions of the frontal lobe are the precentral sulcus, precentral gyrus, and the superior and inferior frontal sulci, which delineate the superior, middle, and inferior frontal gyri. The frontal lobe has 5 functional regions: primary motor cortex, premotor cortex, the frontal eye field, Broca's area, and the prefrontal cortex. The motor homunculus is a visual representation of the areas of primary motor cortex dedicated to the voluntary motor control of body parts. The parietal lobe contains the postcentral sulcus and intraparietal sulcus that border the postcentral gyrus, superior parietal lobule, and inferior parietal lobule. Its functional regions are the primary somatosensory cortex, the somatosensory association cortex, and the supramarginal and angular gyri that both contribute to Wernicke's area. The sensory homunculus is a visual representation of the proportion of sensory axons the primary somatosensory cortex allocates to a particular part of the body. The temporal lobe contains the transverse temporal gyrus of Heschel, which includes the primary auditory cortex, and the superior temporal gyrus, that contributes to a part of Wernicke's area. Structures in the medial temporal lobe are important to learning, memory, and emotion. The occipital lobe contains the cuneus and lingual gyrus separated by the calcarine sulcus, and these areas comprise the primary visual cortex. Deep within the lateral fissure is the insula, or fifth lobe. And finally, around the corpus callosum, we have a strip of cortex, the subcallosal gyrus and the cingulate gyrus, that are both part of the limbic system.
The brain and spinal cord are covered by the meninges, which are 3 layers or membranes of connective tissue that not only protect the brain and spinal cord, but also form a framework for vessels and venous sinuses. Just think of this as the brain needing 3 layers of blankets when going to bed at night to make sure it is extra cozy and secure. These 3 layers, from superficial to deep, are the dura mater, arachnoid mater, and pia mater. The dura mater is a tough, thick, fibrous external meningeal layer. Deep to the dura mater is the arachnoid mater. The dura and arachnoid mater are separated from each other by a potential space called the subdural space. Deep to the arachnoid mater is the pia mater. The pia mater is a delicate vascular layer that is intimately adhered to the brain, covering the gyri and extending along different sulci and fissures. Together, the arachnoid and pia mater are collectively known as the leptomeninges. Between the arachnoid mater and the pia mater is the subarachnoid space, also known as the leptomeningeal space, which is a true space between the arachnoid and pia mater, which contains cerebrospinal fluid, or CSF for short, as well as major vessels and cranial nerves. Okay, let's dive in and take a closer look at the dura mater, which is the thickest, outermost meningeal layer. The dura mater itself is further divided into 2 layers. The first, more superficial layer is called the endosteal layer, or periosteal layer, of the dura mater. It is located on the interior surface of the skull bones and ends at the foramen magnum. The endosteal layer does not continue with the dura mater of the spinal cord, but instead becomes continuous with the periosteum on the external aspect of the skull bones. Between the endosteal layer of the dura mater and the skull bones, there lies a potential space called the extradural or epidural space, which is not a natural space, but may become a pathological space during bleeding. The second deeper layer of the dura mater is the inner meningeal layer, which is continuous with the dura mater of the spinal cord and ends at the level of the S2 vertebra. For the most part, these two dural layers are fused together and cannot be separated. But there are two main exceptions to this. One, there are locations where spaces are created between the layers to house the dural venous sinuses. Secondly, the inner meningeal layer of the dura mater reflects away from the endosteal layer at certain regions to create dural infoldings, or reflections, which divide the cranial cavity into compartments. These dural infoldings, or reflections, consist of the falx cerebri, falx cerebelli, tentorium cerebelli, and diaphragma celli. The largest dural infolding is the falx cerebri, which lies in the longitudinal fissure and separates the two cerebral hemispheres from each other. It attaches anteriorly to the frontal crest of the frontal bone and the crista galli of the ethmoid bone. It contains the superior sagittal sinus in its fixed superior margin and attaches posteriorly to the internal occipital protuberance, where it also blends with the upper surface of the tentorium cerebelli. Speaking of the tentorium cerebelli, this crescent-shaped reflection is the second largest dural infolding, with a fixed margin posterolaterally and a free margin more anteriorly. The fixed margin is attached at three sites: bilaterally at the posterior clinoid processes, to the superior parts of the petrous portion of the temporal bone, and to the grooves of the transverse sinus on the inner surface of the occipital bone. In contrast, the anterior part of the tentorium cerebelli is connected to the clinoid process of the sphenoid bone at its most rostral end. Otherwise, its margins are free, forming a U-shaped space. This space between the free margins of the tentorium cerebelli is called the tentorial notch, and it allows for passage of the brainstem. The tentorium cerebelli spans a transverse plane over the cerebellum, which forms a roof over the posterior cranial fossa. This effectively separates the cerebellum from the cerebrum and divides the cranial vault into an infratentorial compartment below the tentorium cerebelli and a supratentorial compartment above. As you can see, the falx cerebri blends with the tentorium cerebelli posteriorly, helping to maintain its position. Now, inferior to the tentorium cerebelli, we will find the falx cerebelli. This small dural infolding attaches to the internal occipital crest and contains the occipital sinus in its fixed posterior margin. It extends in the sagittal plane and partially separates the cerebellum into two cerebellar hemispheres. The diaphragma celli is the last and smallest dural infolding. It is a flat layer between the clinoid processes that forms an incomplete roof over the hypophyseal fossa of the sella turcica, which is a part of the sphenoid bone and contains the pituitary gland. The diaphragma celli has a circular opening in the middle, which allows the passage of the pituitary stalk, or infundibulum, to connect the hypothalamus above to the pituitary gland below. Now, as we said before, the two layers of the dura mater, for the most part, are strictly fused together. However, there are areas between the endosteal and meningeal layers that form spaces to accommodate the dural venous sinuses. These dural venous sinuses contain venous blood from the cerebral veins and also cerebral spinal fluid from the subarachnoid space. The cerebrospinal fluid enters the sinuses through structures called arachnoid granulations, which protrude through the meningeal dura mater into the dural venous sinuses. The contents of the dural venous sinuses ultimately drain into the internal jugular vein. The dural venous sinuses can be either paired or unpaired. Paired sinuses include the transverse sinus, cavernous sinus, superior petrosal sinus, inferior petrosal sinus, sphenoparietal sinus, and sigmoid sinus. On the other hand, unpaired sinuses include the superior sagittal sinus, inferior sagittal sinus, straight sinus, occipital sinus, and intercavernous sinus. The superior sagittal sinus is the largest dural venous sinus, and throughout its course, it receives venous blood from the superior cerebral veins. As the name suggests, it runs in the sagittal plane along the border of the falx cerebri. The superior sagittal sinus extends from the foramen cecum of the frontal bone rostrally to the internal occipital protuberance caudally, where it drains into the confluence of sinuses. Keep in mind that the confluence of sinuses is also the point where the straight and occipital sinuses merge with the superior sagittal sinus. It then deviates to one side, usually the right, to connect with the transverse sinus. The left transverse sinus and right transverse sinus begin at the internal occipital protuberance from the confluence of sinuses and pass laterally to run in the lateral border of the tentorium cerebelli. While doing so, these sinuses create impressions in the occipital and parietal bones. The transverse sinuses also receive blood from the superior petrosal sinuses. The two transverse sinuses eventually continue as the right and left sigmoid sinuses on each side. The right and left sigmoid sinuses follow an S-shaped course in the posterior cranial fossa. During their course, they turn anteriorly and continue inferiorly as the internal jugular veins, which travel through the jugular foramen. Deeper to the superior sagittal sinus lies the smaller, unpaired inferior sagittal sinus, which runs along the inferior free border of the falx cerebri. Similar to the superior sagittal, it has a rostral to caudal extent. During its course, it receives venous blood from small veins draining the medial surface of the cerebral hemispheres and ultimately drains into the straight sinus. The straight sinus is formed by the merger of the inferior sagittal sinus and the great cerebral vein. The straight sinus runs postero-inferiorly along the attachment between the falx cerebri and the tentorium cerebelli to eventually meet the superior sagittal and occipital sinuses at the confluence of sinuses. Speaking of the occipital sinus, this sinus is located on the anterior aspect of the occipital bone, along the caudal attached border of the falx cerebelli. It ends posteo-superiorly at the confluence of sinuses. Up next, we have the cavernous sinuses, which are located within the middle cranial fossa on either side of the sella turcica of the sphenoid bone. The cavernous sinus extends from the superior orbital fissure to the apex of the petrous part of the temporal bone. The left and right cavernous sinuses are connected to each other via intercavernous sinuses, which are quite variable but usually consist of anterior and posterior parts. The cavernous sinuses then diverge into the superior petrosal sinus and the inferior petrosal sinus. The superior petrosal sinus drains into the transverse sinus at the site where it continues as the sigmoid sinus, and the inferior petrosal sinus drains into the sigmoid sinus at its transition to becoming the internal jugular vein. Off note, there are bridging vessels called emissary veins that connect the dural venous within the cranium to veins outside of the cranium. One example is the parietal emissary vein, which runs through the parietal foramen and connects the superior sagittal sinus with veins in the scalp. Okay, that was a lot. Let's take a short break and see if you can recall these dural venous sinuses. Of all the sinuses that we have talked about, the cavernous sinus is the most clinically relevant because there are several important structures related to it. Again, the cavernous sinus lies on both sides of the sella turcica of the sphenoid bone. The pituitary gland lies in between the sinuses, along the midline, within the hypophyseal fossa. Looking at a coronal section, the cavernous sinus contains numerous fibrous trabeculi that result in the sinus having a spongy, cave-like appearance. The cavernous sinus has a medial and lateral border, a floor, and a roof. The medial border of the cavernous sinus is formed by the sphenoid bone, and the lateral border is formed by the meningeal layer of the dura mater. The roof of the cavernous sinus is the diaphragma celli, which is also the meningeal layer of the dura mater. And lastly, the floor is formed by the endosteal layer of the dura mater overlying the sphenoid bone. There are several key anatomical structures that travel either through the cavernous sinus or very close to it. The structures passing through the cavernous sinus are the internal carotid artery and the postganglionic sympathetic fibers of the carotid plexus that run with it. as well as the abducens nerve, or cranial nerve 6. In contrast to running through the cavernous sinus, the ocular motor nerve, or cranial nerve 3, the trochlear nerve, or cranial nerve 4, and the ophthalmic, or V1, and maxillary, or V2 branches of the trigeminal nerve, or cranial nerve 5, travel along the lateral wall of the sinus. Now, the dura mater is highly vascularized with its own intricate arterial circulation. The major artery that supplies the dura is the middle meningeal artery, which is a branch of the maxillary artery. It passes through the skull in the foramen spinosum and enters the middle cranial fossa. From here, the middle meningeal artery runs laterally and then superiorly on the greater wing of the sphenoid bone, where it divides into the frontal or anterior branch and the parietal or posterior branch. The parietal branch runs posteriorly and superiorly to supply the posterior part of the dura and cranium. On the other hand, the frontal branch heads superiorly to the pterion and eventually runs posteriorly to give off several branches that distribute throughout the dura up to the vertex of the skull. The dura mater also has an extensive nerve supply, which directly or indirectly comes from the trigeminal nerve, or cranial nerve 5. The dura overlying the anterior cranial fossa is innervated by branches of all three divisions of the trigeminal nerve. The dura overlying the middle cranial fossa is innervated mainly by branches of the mandibular nerve, or V3, with some innervation from the maxillary nerve, or V2. Furthermore, the supratentorial dura covering the posterior cranial fossa is innervated by branches of the ophthalmic nerve, or V1. The infratentorial dura is innervated by branches of the cervical plexus arising from C2 and C3, as well as the vagus nerve, or cranial nerve 10. Pain signals from the dura travel along the sensory fibers of these nerves and can be referred to the cutaneous areas that are also supplied by the same nerve fibers. For example, irritation of the dura from an infection in the anterior cranial fossa can be referred to any of the cutaneous areas that are also supplied by those same branches of the trigeminal nerve, such as the skin around the orbit or face. Okay, let's move on to the two layers that form the leptomeninges, the arachnoid mater and the pia mater. The arachnoid mater is situated between the dura and pia mater, creating two spaces. The first is the subdural space, which is a potential space between the dura and arachnoid mater. The second is the subarachnoid space between the arachnoid and pia mater, which is filled with CSF, as well as cerebral arteries, veins, and cranial nerves. The arachnoid mater forms protrusions called arachnoid granulations, which pierce the dura mater to enter the dural venous sinuses. These protrusions allow the CSF to flow from the subarachnoid space into the dural venous sinuses, and from there into the venous circulation. The largest granulations are present along the length of the superior sagittal sinus, located just below the calvaria. These granulations may erode into the inner surface of the skull, creating small pits known as the granular foveolae. The arachnoid mater is also loosely connected to the pia mater through thin strands of fibrous tissue known as arachnoid trabeculae. These strands create a meshwork that looks similar to a spider web, hence the name arachnoid for these structures. Where the various cranial nerves exit the skull, the arachnoid mater fuses with their outside layer called the epineurium. Finally, we have the pia mater, which is the deepest meningeal layer, and we don't mean the most profound. This is the most delicate and the thinnest meningeal layer, and it tightly adheres to the entire surface of the brain. The cranial pia mater is the only meningeal layer that closely follows the contours of the brain. The pia mater dives into all of the sulci and fissures, blending for a short time with the sheaths of arteries as they enter the brain, and fuses with the epineurium of the cranial nerves within the subarachnoid space. In this video, we have focused on the cranial meninges. However, please note that there are slight differences between the cranial meninges and the meninges surrounding the spinal cord. Basically, the dura mater of the cranial meninges is double-layered with both endosteal and meningeal layers, while the spinal meninges only have a single meningeal layer. Also, the epidural space, in relation to the cranial meninges, is a potential space, while the epidural space superior to the dura of the spinal cord is a real space. Alright, as a quick recap, there are three meningeal layers that consist of membranous connective tissue that covers the brain and the spinal cord, The outermost and toughest layer is the dura mater, which has two layers, the outer endosteal and the inner meningeal layer. The endosteal layer is an extension of the periosteal layer of the anterior skull bones, whereas the deeper meningeal layer creates projections that form dural infoldings or reflections. The arachnoid mater is found deep to the dura mater, and between them is the subdural space. The arachnoid mater contains arachnoid granulations, which pierce the dura mater to enter the dural venous sinuses. The deepest meningeal layer is the pia mater, which closely adheres to the contours of the brain. The arachnoid mater is loosely connected to the pia mater through the arachnoid trabeculae. Between these two layers is the subarachnoid space, which contains CSF, cerebral vessels, and cranial nerves. The main dural infoldings are the falx cerebri, the tentorium cerebelli, the falx cerebelli, and the diaphragma celli. These two dura mater layers are normally attached to each other. However, there are areas where pockets occur between them, forming the dural venous sinuses that drain cerebral venous blood and cerebrospinal fluid. The dura has extensive sensory innervation from different branches of the trigeminal nerve, as well as the vagus nerve and branches from cervical nerves C2 and C3.
The limbic system consists of several parts within the telencephalon, also known as the cerebrum, the diencephalon, which includes the thalamus and hypothalamus, and the midbrain. The term limbic means border in Latin, so this system's name originated from its location at the border between the telencephalon and diencephalon. One way to remember the limbic system is to think of the word home, homeostasis, olfaction, memory, emotion. This is because limbic system structures are involved in olfaction, or smell, in the regulation of emotions, like anger and fear, and other behaviors, like aggression and sexual behavior. Memory formation and the recollection of those memories are supported by the limbic system, and it can even influence responses of the autonomic nervous system, like cardiovascular or gastrointestinal functions. To easily recall some of the functions, you can remember the famous 5 F's, which are feeding, fleeing, fighting, feeling, and fornicating, the last one being really just a fancy word for sex. Structures that are included in the limbic system include the hypothalamus, olfactory cortex, hippocampal formation, amygdala, subcallosal area, cingulate gyrus, parahippocampal gyrus, mammillary bodies, and the basal forebrain. Let's take a closer look at each of them, starting with the hippocampal formation, which is crucial for converting short-term memory into long-term memory, shipping those memories into other cortical regions for long-term storage, and assisting in retrieval of memories when needed. It is also involved in spatial orientation, which is the ability to identify the position of our body relative to the objects around us. The hippocampal formation consists of the hippocampus, the dentate gyrus, subiculum, and entorhinal cortex. Now, on a mid-sagittal section of the brain, the hippocampal formation is hidden within the medial temporal lobe. It lies posterior to the amygdala and spreads caudally all the way to the splenium of the corpus callosum. On a transverse section of the temporal lobe, we can see the hippocampal formation forming the floor of the inferior horn of the lateral ventricle. But the best plane for the visualization of this structure is actually the coronal plane. Here, we can see the hippocampal formation located within the medial temporal lobe and swooping beneath the inferior horn of the lateral ventricle. The most inferior part of the hippocampal formation is the subiculum, and it is continuous caudally with the parahippocampal gyrus. The hippocampus, meaning seahorse in Latin, is so named as its contours resemble the shape of a seahorse. The dentate gyrus is contained within the hippocampus, just superior to the subiculum. The hippocampus is involved in long-term memory formation and helps decide what should be stored within the cerebral cortex as a memory. It has connections with the olfactory cortex, hence why smells can elicit memories very easily, and also connections with the amygdala to remember emotionally significant events. The next three structures that belong to the limbic system are the subcallosal area, the cingulate gyrus, and the parahippocampal gyrus. These structures influence emotional reactions and support learning and memory formation. On a mid-sagittal section of the brain, we can see the subcallosal area on the medial side of the frontal lobe, just anterior to the lamina terminalis and inferior to the rostrum of the corpus callosum. Anteriorly, it is continuous with the cingulate gyrus, which arches over the genu, body, and splenium of the corpus callosum. Superior to the cingulate gyrus, there is the cingulate sulcus that separates it from the rest of the cerebral cortex. On a coronal section of the brain, we can see the corpus callosum connecting the hemispheres and the cingulate gyrus just superior to it at the bottom of the longitudinal fissure. On the ventral and medial aspect of the temporal lobe, spreading anteriorly from the splenium of the corpus callosum, we see the parahippocampal gyrus, which is separated from the rest of the temporal lobe by the collateral sulcus. The anterior portion of the parahippocampal gyrus curves medially and posteriorly like a hook and is called the uncus. And now let's look at the mammillary bodies, which are a pair of round nuclei belonging to the hypothalamus that can be seen on the ventral surface of the brain. They are located posterior to the tuber cinereum of the hypothalamus, anterior to the midbrain, and between the cerebral peduncles. Similarly, in the mid-sagittal plane of the brain, they can be seen posterior to the tuber cinereum and anterior to the tegmentum of the midbrain. In the coronal plane, we can see the mammillary bodies inferior to the third ventricle. Now, all of the hypothalamic nuclei are part of the limbic system, as homeostasis is one of their major functions, but the mammillary bodies are especially important, as they play a role in learning and memory and are connected to the hippocampus. These structures assist in episodic memory, which is the ability to recall specific situations, or episodes, from our lives, like the place and time of our first kiss and the emotional reaction associated with it. Great! Now let's take a closer look at the amygdala, which means almond in Latin. The amygdala contains numerous nuclei and is connected with various parts of the nervous system, but the important connections with other limbic system structures include the hypothalamus, olfactory cortex, hippocampal formation, frontal lobe, and the sensory association cortices. Because of this, the amygdala is involved in emotional responses, especially fear, rage and aggression, modulation of memory, and influences attention and decision-making. Interestingly, the connections between the amygdala and the hypothalamus can elicit various physiological responses to emotional experiences. On a mid-sagittal section of the brain, the amygdala is located in the medial temporal lobe, just deep to the uncus. On a rostral coronal plane, we can also see the amygdala in the medial temporal lobe, deep to the uncus with the tail of the caudate just superior to it. Remember that the hippocampus and parahippocampal gyrus would be found more caudally in relation to the amygdala. Let's take a short break and see if you can recall the role of the hippocampal formation. Okay. Now that we saw the primary structures that make up the limbic system, let's go through just a few more structures that are connected to and work together with it. First, there's the orbitofrontal cortex of the frontal lobe, which is involved in planning and decision-making in relation to emotion and reward. It's connected with the dorsomedial nucleus of the thalamus, which gathers information from other cortical and limbic system structures like the hypothalamus and amygdala. It relays this information to the orbitofrontal cortex to aid in decision-making, reward learning, affective behavior, emotional reactions, subjective feeling states, and personality. Another part of the thalamus that is connected to the limbic system includes the anterior nuclei of the thalamus. They receive input from the mammillary bodies and project to the cingulate gyrus, supporting the pathway for long-term memory storage. Two more groups of structures that are connected to the limbic system and the basal ganglia include the basal forebrain nuclei and limbic nuclei in the brainstem. The basal forebrain nuclei include the basal nucleus of Meinert, the nucleus accumbens, and the septal nuclei. On a coronal section of the brain, the basal nucleus of Meinert can be seen just below the basal ganglia. It contains neurons that release acetylcholine as their neurotransmitter. The nucleus accumbens can be seen within the basal ganglia, next to the septal area. It contains neurons that release GABA as their neurotransmitter. The septal nuclei are believed to be a pleasure area of the brain involved in reward and reinforcement. These nuclei are part of the basal forebrain and are connected to other limbic system structures such as the amygdala, the olfactory cortex, the hippocampus, and the hypothalamus. In the mid-sagittal plane, we see them located inferior to the rostrum of the corpus callosum, superior to the lamina terminalis, and around the anterior commissure. Moving on to the limbic nuclei in the brainstem, these nuclei include the ventral tegmental area, the raphe nuclei, and the locus ceruleus. The ventral tegmental area is located in the ventral midbrain, medial to the substantia nigra, and its neurons are dopaminergic. The raphe nuclei can be found throughout the brainstem as part of the reticular formation, and their neurons produce serotonin. The locus ceruleus is located in the pons and contains norepinephrine-producing neurons. All of these act as diffuse modulators within the nervous system. Let's take another short break and see if you can recall the parts of the limbic system. Lastly, let's cover a few of the axon tracts that connect the parts of the limbic system and allow for overall communication and function. Although connections are rich and varied, a few of the major tracts include the fornix, the mammillothalamic tract, and the stria terminalis. The fornix is made up of fibers that run from the hippocampus to the mammillary bodies, and it has several parts. First, there are the fimbria, which are fibers that arise from the hippocampus. They gather and leave the hippocampus posteriorly as the crus of the fornix. The left and right crura of the fornix curve posteriorly and then superiorly, passing posterior to the thalamus and inferior to the splenium of the corpus callosum. They come together to form the body of the fornix, which lies beneath the corpus callosum and superior in relation to the third ventricle. Before they merge into the body, the crura are connected by a membrane called the commissure of the fornix, or the hippocampal commissure. It contains fibers that connect the left and right hippocampi. The body of the fornix then curves anteriorly and inferiorly with the septum pellucidum located superior to it. The septum pellucidum is a membrane between the lateral ventricles, and on a medial view we see it extending between the fornix and the corpus callosum. Near the anterior commissure, the body of the fornix splits into two columns of the fornix. These pass over the interventricular foramina and pass through the hypothalamus before reaching the mammillary bodies where it will terminate. The second major bundle of axons is the mammillothalamic tract that connects the mammillary bodies with the anterior nuclei of the thalamus. The last pathway is the stria terminalis, which connects the amygdala with the basal forebrain and the hypothalamus. The fibers of the stria terminalis originate in the amygdala and pass along the medial side of the tail of the caudate nucleus, following it through the floor of the body of the lateral ventricle before reaching the hypothalamus. Alright, as a quick recap, The limbic system consists of several parts of the telencephalon, diencephalon, and midbrain that together influence emotions, behavior and learning, and memory. The hippocampal formation consists of the hippocampus, dentate gyrus, subiculum, and entorhinal cortex, and it's involved in long-term memory formation and spatial orientation. These structures, along with the mammillary bodies, help to support the consolidation and storage of memories. Then there is the subcallosal area, the cingulate gyrus, and the parahippocampal gyrus, which support emotional responses and learning and memory. The amygdala regulates emotions like fear and aggression and can elicit physiological responses to emotion via its connections with the hypothalamus. Then there are areas that are functionally connected to the limbic system, and these include the orbitofrontal cortex and the dorsomedial nuclei and anterior nuclei of the thalamus. The basal forebrain includes the basal nucleus of Meinert, the nucleus accumbens, and the septal nuclei. And finally, the limbic midbrain area that includes the ventral tegmental area, which produces dopamine, the raphe nuclei, which produces serotonin, and the locus ceruleus, which produces norepinephrine. The major pathways that connect the structures of the limbic system include the fornix, the mammillothalamic tract, and the stria terminalis.
An adult human brain weighs about 1.5 kilograms, but we don't really feel it weighing us down. That's because the brain is cushioned by cerebrospinal fluid, or CSF, which can be found within brain cavities called ventricles. These cavities are involved in the production, transport, and removal of CSF, and they are connected to each other. So as a whole, they are often referred to as the ventricular system of the brain. CSF doesn't only fill the ventricles, but also the subarachnoid space, which surrounds the brain and spinal cord. This way, CSF cushions and protects the brain from head trauma, and it also provides buoyancy, so that the brain doesn't compress blood vessels and cranial nerve roots against the cranium. It also provides protection against sudden intracranial pressure changes. CSF can also transport nutrients for nervous tissue, as well as remove metabolic waste products. It can also transport hormones and influence the brain's excitability by regulating its ionic composition. Now, the ventricular system consists of 4 ventricles, 2 lateral ventricles, the 3rd ventricle, and the 4th ventricle. CSF flows from the lateral ventricles through the left and right interventricular foramina, also called the foramina of Monro, to the 3rd ventricle. From here, it passes through the cerebral aqueduct to reach the 4th ventricle. CSF can then flow caudally into the central canal of the spinal cord. The 4th ventricle also has 2 lateral apertures, called the foramina of Luschka, and a median aperture, called the foramen of Magendie, both of which allow CSF to reach the subarachnoid space. The lateral ventricles are the largest. They occupy both cerebral hemispheres, and they're present in all 4 lobes. This is best seen on coronal sections of the brain, cutting through the frontal lobe, the parietal lobe, and temporal lobes, and finally the occipital lobe. On a mid-sagittal section of the brain, you can see the 3rd ventricle centrally. Rostrally and superiorly, there is a depression that narrows into the interventricular foramen, which connects the 3rd ventricle to the 2 lateral ventricles. Inferiorly, the 3rd ventricle continues into the cerebral aqueduct, which is a narrow canal that passes through the midbrain and reaches the fourth ventricle, which is a pyramid-like cavity sitting dorsal to the brainstem and ventral to the cerebellum. Caudally, the fourth ventricle continues to the central canal of the spinal cord, and just inferior to the cerebellum, there is the foramen of Magendie. Laterally, at the level of the cerebellum, there are two foramina of Luschka. These foramina allow CSF to leave the fourth ventricle and enter the subarachnoid space. The subarachnoid space has CSF-filled dilations called subarachnoid cisterns. Some of these cisterns contain proximal parts of cranial nerves and blood vessels. On a mid-sagittal section of the brain, we can identify the main subarachnoid cisterns. Between the cerebellum and the medulla, there is the posterior cerebellomedullary cistern, also known as cisterna magna. It receives CSF from the foramen of Magendie, and can even be accessed for obtaining a CSF sample in some rare cases. On both sides of the cisterna magna, there are two lateral cerebellomedullary cisterns that can't be seen in this section. They receive CSF from the foramina of Luschka and contain cranial nerves 7 and 8. Then, ventral to the pons, there's the pontocerebellar, or pontine cistern, which contains the basilar artery and continues inferiorly into the spinal subarachnoid space. Next is the interpeduncular, or basilar cistern, which sits between the cerebral peduncles of the midbrain. Rostrally, there's the chiasmatic cistern, or the cistern of the optic chiasma, which lies below the optic chiasm. And lastly, the quadrigeminal cistern sits inferior to the caudal part of the corpus callosum and superior to the cerebellum. It is also referred to as the cistern of the great cerebral vein, since it contains the great cerebral vein of Galen. No surprises here. Next, let's go over the choroid plexus. Now, CSF is produced by the epithelial cells of the choroid plexus, contained in all four ventricles. The choroid plexus is a cauliflower-like structure that consists of many fringes and folds of vascular pia mater that protrude into the ventricles. And surrounding the pia mater, there are cuboidal epithelial cells. These are actually modified ependymal cells, similar to those that line the inner surface of the ventricles. The choroid plexus can be found protruding from the floor of the lateral ventricles and the roof of the third and fourth ventricles. As CSF flows from the ventricles to the subarachnoid space, it continues superiorly to reach the superior sagittal sinus, which is a channel between two layers of dura mater that holds venous blood. The arachnoid mater forms small protrusions, called arachnoid villi, that penetrate the dura and enter the sinus. The subarachnoid space extends into these villi, which are therefore filled with CSF. The CSF then passes through the thin lining of the villi and drains into the venous system to be recycled. Interestingly, the villi tend to group together and form arachnoid granulations. You can even see the impressions of these granulations, called the granular foveolae, on the inferior surface of the calvaria. Okay, now let's look at each ventricle in a bit more detail. First up, the lateral ventricles. Each of them is shaped like the letter C, and they each have a body, or a central part, and 3 horns. The anterior horn, posterior horn, and inferior horn, also called the frontal, occipital, and temporal horns respectively. To begin, there's the body of the lateral ventricle, and it can be found deep within the parietal lobe. The body of the lateral ventricle has a distinct roof, a floor, and a medial wall. On a coronal section of the brain, you can see that the roof is formed by the inferior surface of the corpus callosum. The floor, on the other hand, from medial to lateral, is formed by the fornix, choroid plexus, lateral part of the dorsal side of the thalamus, and the caudate nucleus. Medially, the septum pellucidum extends from the corpus callosum to the fornix, separating the left and right lateral ventricles. On more caudal coronal sections, the septum pellucidum slowly disappears as the corpus callosum and fornix come closer together. Rostrally, the body connects to the interventricular foramina, which can be seen on a transverse section of the brain. Each interventricular foramen is bounded by the anterior column of the fornix rostrally and the anterior aspect of the thalamus caudally. As we follow the body of the lateral ventricle caudally, we see that it stretches along the medial aspect of the body of the caudate nucleus, and eventually becomes continuous with the posterior and inferior horns. On a mid-sagittal section of the brain, we can easily identify the corpus callosum and the septum pellucidum, extending from it to the fornix. Caudally, the septum pellucidum becomes smaller as the roof and the floor of the lateral ventricles come closer together. If we look at a three-quarter view of the brain and remove the cerebrum, we can see how the body of the lateral ventricle curves around the thalamus and caudate nucleus, and the corpus callosum is located superior to it. Rostrally, the body is continuous with the anterior horn. Now, the anterior horn continues from the body at the interventricular foramen and extends into the frontal lobe. On a coronal section, we can see that the anterior horn has a roof, a floor, and a medial wall. The roof is also formed by the corpus callosum, while the floor is formed by the head of the caudate nucleus. The medial wall is formed by the septum pellucidum and the anterior columns of the fornix. Again, if we look at a three-quarter view of the brain and remove the cerebrum, we can appreciate the relationship between the lateral ventricle and the basal ganglia. Here, we can see how the anterior horn is above the head of the caudate nucleus and below the corpus callosum. As the rostral part of the corpus callosum curves inferiorly and caudally, it also forms the rostral border of the anterior horn, and even a small portion of its floor. On a transverse section at the level of the interventricular foramina, we can follow the anterior horn from the foramen along the medial side of the head of the caudate nucleus. The posterior horn continues from the body at the posterior end of the thalamus and extends into the occipital lobe. The posterior horn has a roof, or a lateral wall, and a floor, or a medial wall. On a coronal section, you can see the roof is formed by the fibers spreading laterally from the corpus callosum, called the tapetum. Lateral to the tapetum, there are fibers of the optic radiation. The floor has a superior and inferior bump. The superior bump, also known as the bulb of the posterior horn, is formed by the fibers of the corpus callosum, called the forceps major, spreading posteriorly. The inferior bump, also known as the calcar avus, is formed by the underlying calcarine sulcus. On a transverse section, the optic radiation borders the posterior horn laterally. And on a mid-sagittal section, if we ghost the ventricles, we can see how the calcarine sulcus reaches the posterior horn. Finally, the inferior horn continues from the body inferiorly, and then rostrally extending into the temporal lobe. This horn also has a roof and a floor, both of which are visible on a coronal section. The roof is formed by the tapetum, spreading laterally from the corpus callosum. On the medial and superior aspect, there's the tail of the caudate nucleus. The lateral part of the floor is formed by the collateral eminence, which is an elevation caused by the collateral sulcus underneath it. The medial part of the floor is formed by the hippocampus. The ventricular surface of the hippocampus is lined by the alveus, which is formed by the fibers of the neurons inside the hippocampal cortex. Fibers of the alveus group together to form the fimbria, which are positioned medial to the hippocampus. On a transverse section, the hippocampus extends along the medial part of the floor of the inferior horn. Rostrally, it expands and becomes a bit wrinkly, forming the pes of the hippocampus. Looking at a three-quarter view, we can see that the inferior horn is bordered rostrally by the amygdaloid nucleus. Also, the tail of the caudate nucleus on the roof extends all the way to the amygdala as well. Okay, time for a quick break. Can you remember the names of the three horns of the lateral ventricles? Great! Now let's look at the third ventricle, which is a very narrow cavity in the middle of the diencephalon. On a mid-sagittal section, the superior part of the lateral wall is formed by the thalamus, while the inferior part is formed by the hypothalamus. The third ventricle is bordered rostrally by the anterior commissure and the lamina terminalis. On the anterior and superior part of the lateral wall, there's the interventricular foramen, while in the middle, there's the interthalamic adhesion, which is a bridge of tissue connecting the two thalami. The third ventricle also has small extensions that form recesses near adjacent structures. These include the optic recess, between the lamina terminalis and the optic chiasm, the infundibular recess, towards the pituitary gland, the pineal recess, towards the pineal gland, and the suprapineal recess, above the epithalamus. On a coronal section, you can see that the roof is formed by the choroid plexus, the floor is formed by the hypothalamus, and laterally there are the thalami. On a transverse section, the third ventricle looks like a slit, or cavity between the thalami, that continues into the lateral ventricles at the level of the interventricular foramen. Now on a mid-sagittal section, the third ventricle connects inferiorly and caudally with the cerebral aqueduct, or the aqueduct of Sylvius. The cerebral aqueduct stretches through the dorsal part of the midbrain, inferiorly towards the fourth ventricle. This is the narrowest passage of the ventricular system that the CSF flows through, and it doesn't usually have choroid plexus. On a coronal section, we can see it as a central opening, or canal, extending down the brainstem. On a transverse section of the midbrain, the cerebral aqueduct is positioned in the middle, between the tectum posteriorly and the tegmentum anteriorly. Surrounding it is the periaqueductal gray matter. Lastly, let's cover the fourth ventricle. It has a dorsal wall, or a roof. On a mid-sagittal section, the fourth ventricle looks like a tent. Its roof has a central part called the apex, or the fastigium, that extends towards the cerebellum. From the apex, the superior medullary velum extends superiorly. This is a thin layer of white matter between the superior cerebellar peduncles. There's also an inferior medullary velum that extends from the apex inferiorly. This structure holds the choroid plexus of the fourth ventricle, and it also contains the foramen of Magendie in the midline, where most of the CSF leaves the fourth ventricle into the subarachnoid space. The ventral wall, or floor of the fourth ventricle, on the other hand, is formed by the dorsal sides of the pons and the medulla. In the midline, the median sulcus can be seen, extending from the cerebral aqueduct rostrally to the central canal of the spinal cord caudally. To better visualize the floor of the fourth ventricle, we should expose the dorsal surface of the brainstem. The floor is diamond or rhomboid shaped, so it's called the rhomboid fossa. The dorsal aspect of the pons, which is triangular shaped, creates the rostral half of the floor. Laterally, there are the superior cerebellar peduncles. Running vertically down the midline is the median sulcus, and just parallel to it, on each side, the sulcus limitans. Between the median sulcus and sulcus limitans, there are two elevations called the medial eminences, whose caudal parts enlarge to form the facial colliculi. The facial colliculi are two bumps, which are made up of axons from the facial nuclei that wrap around the abducens nuclei. Caudal to the superior cerebellar peduncles, we see the middle cerebellar peduncles. Now, on the dorsal aspect of the medulla oblongata, we can see that the rostral medulla creates the dorsal half of the floor of the fourth ventricle. Laterally, there are the inferior cerebellar peduncles, which contain fibers that travel between the medulla and the cerebellum. The ventricle, or fossa, tapers at its caudal aspect to a point called the obex. And near this caudal limit is the entrance to the central canal of the spinal cord. In this region, CSF can travel from the fourth ventricle into the central canal. Lastly, let's look at the area postrema, which can be found on the dorsal surface of the medulla on the floor of the fourth ventricle, just before the entrance to the central canal. It holds the chemoreceptor trigger zone, or CTZ, that can initiate the vomiting reflex by sending input to the vomiting center in the medulla. The CTZ doesn't have a well-developed blood-brain barrier, which means that it can be exposed to chemical substances found in the blood and CSF. These substances include toxins, hormones, alcohol, and drugs, such as chemotherapy drugs. The substances can in turn directly activate and initiate the vomiting reflex. It also receives input from visceral afferent fibers coming from the GI tract via the vagus nerve. For example, chemotherapy drugs can stimulate receptors on vagal afferent nerve fibers in the bowel wall of the gastrointestinal tract, which ascend and trigger the vomiting reflex in the CTZ. Additionally, it also receives input from the vestibular system, which can explain car or seasickness. All right, as a quick recap, the ventricular system produces CSF via the choroid plexuses and transports it to the subarachnoid space. The lateral ventricles have a body in the parietal lobe and three horns, the anterior, posterior, and inferior horns, that extend into the frontal, occipital, and temporal lobes respectively. The CSF flows from the lateral ventricles through the interventricular foramina of Monro to the third ventricle that is located in the middle of the diencephalon. The CSF then flows through the cerebral aqueduct of Sylvius of the midbrain into the fourth ventricle, found between the brainstem and the cerebellum. The CSF then enters the central canal of the spinal cord, or the subarachnoid space, through the median foramen of Magendie and two lateral foramina of Luschka. From the subarachnoid space, the CSF gets recycled into the venous system via the arachnoid villi and granulations.
Our central nervous system consists of three major parts: the cerebrum, the cerebellum, and the brainstem. The largest part is the cerebrum, which consists of two nearly symmetrical halves called the cerebral hemispheres. If we were to cut through the cerebral hemispheres in the coronal plane, we would see that the outermost area is the cerebral cortex, which consists of gray matter that contains billions of neuronal cell bodies. Deep to the gray matter is the subcortical white matter, which is made up of myelinated axons that are extensions of the neuronal cell bodies, allowing them to send and receive signals. The term white matter is used because the myelination of the axon fibers gives this area a white appearance on gross inspection. Axon fibers can be divided into three main groups: commissural fibers, association fibers, and projection fibers, depending on the target of the axons. The cerebral commissural fibers connect the left and right cerebral hemispheres and consist of the corpus callosum, the anterior commissure, the posterior commissure, and the hippocampal commissure. Association fibers connect different regions within the same hemisphere, and they include the U fibers, uncinate fasciculus, cingulum bundle, arcuate fasciculus, superior longitudinal and inferior longitudinal fasciculi, and the occipitofrontal fasciculus. Finally, projection fibers connect the cerebral cortex with more caudal structures in the central nervous system, like the thalamus, brainstem, and spinal cord. These ascending and descending projection fibers all travel in a structure called the internal capsule, which has 5 parts that we will explore later. Okay, first, let's cover the cerebral commissural fibers. Starting with the corpus callosum, which means tough body in Latin, this is the largest white matter structure in the brain. It sends signals between the two cerebral hemispheres and connects them together at the same time. If we make a mid-sagittal section through the brain, we can see that the corpus callosum has 4 parts: the rostrum, genu, body, and splenium. The body is the central and longest part of the corpus callosum and continues rostrally as the genu and caudally as the splenium. The body of the corpus callosum occupies the bottom of the longitudinal fissure and arches between its rostral and caudal extents. The genu, meaning knee, is the rostral bend of the corpus callosum that curves in front of the septum pellucidum, which is a thin membrane that separates the anterior horns of the lateral ventricles. Just ventral and posterior to the genu is the rostrum, which gets its name because it is similar in shape to a bird's beak. The rostrum connects with the lamina terminalis of the diencephalon. Finally, most caudally, the corpus callosum ends as the splenium, which is an enlarged part of the commissure that sits above the midbrain. Now, let's look at the axial or transverse section of the brain so that we can follow the fibers of the corpus callosum as they fan out towards the cerebral cortex. The fibers from the genu spread rostrally towards the frontal cortex, forming the forceps minor, while the fibers from the splenium spread caudally towards the occipital cortex, forming the forceps major. Lastly, fibers from the body of the corpus callosum spread laterally towards the temporal cortex. A part of this lateral radiation forms a roof over the central part of the lateral ventricle, and hence it is called the tapetum, which means covering or carpet. On a coronal section of the brain, we can see the corpus callosum at the bottom of the longitudinal fissure and superior to the lateral ventricles, forming a bridge between the hemispheres. Next, there is the anterior commissure, which is a much smaller white matter structure that mainly connects the temporal lobes of each hemisphere, but also has some decussating fibers from the olfactory tracts that will terminate in the primary olfactory cortex. On a mid-sagittal section of the brain, the anterior commissure can be observed just posterior to the lamina terminalis and rostral to the column of the fornix. On an axial section of the brain, we can also see the anterior commissure just rostral to the fornix. Now, the fibers of the anterior commissure split laterally into 2 bundles, anterior and posterior. The anterior bundle is smaller and turns ventrally towards the anterior perforated substance and olfactory tracts, while the posterior bundle turns caudally towards the temporal lobe. On a coronal section, we can see the posterior bundle of fibers cutting through the inferior part of the lentiform nuclei while heading towards the temporal lobe. Another commissure to consider is the posterior commissure, which is located between the midbrain and the diencephalon, just rostral to the pineal gland. It is a small commissure and contains axons related to the oculomotor complex, for example, for the pupillary light reflex. Note that the corpus callosum, anterior commissure, and posterior commissure can all be easily seen in the mid-sagittal plane upon gross inspection, or MRI. Lastly, there is the hippocampal commissure, also known as the commissure of the fornix, which connects the left and right hippocampi located in the medial temporal lobes. The fibers from the hippocampi form the posterior columns of the fornix that travel between the thalamus inferiorly and the corpus callosum superiorly. Before merging to form the body of the fornix, the posterior columns are connected by the hippocampal commissure, which is a thin membrane located just inferior to the splenium of the corpus callosum. Let's take a short break and see if you can identify the main commissural fibers and the regions that they connect. Alright then, let's switch gears and talk about the association fibers. These fibers connect different cortical areas within the same cerebral hemisphere, enabling those regions to communicate with each other. Depending on their length, the association fibers can be divided into two groups, the short and long fibers. The short association fibers connect adjacent gyri of the cerebral cortex. They are also known as arcuate fibers, or U fibers, as they resemble a U shape while arching beneath the sulci separating the gyri. On the other hand, the long association fibers connect the cortices of more distant gyri and are grouped into fascicles, or bundles. First, there is the superior longitudinal fasciculus, which connects the frontal, temporal, and occipital lobes. Its fibers pass from the rostral portion of the frontal lobe and through the operculum, which are parts of the frontal, parietal, and temporal lobes that cover the insula. These fibers then reach the caudal aspect of the lateral sulcus and spread to the temporal and occipital lobes. Then there is the fronto-occipital fasciculus, which also connects the frontal, temporal, and occipital lobes. However, its fibers are found in a deeper part of the white matter, passing just lateral to the caudate nucleus. Next is the uncinate fasciculus, which connects the inferior portion of the frontal lobe with the anterior part of the temporal lobe, transversing the bottom of the lateral sulcus. The arcuate fasciculus connects the area of the brain responsible for speech production, or Broca's area, found in the inferior frontal gyrus, with the region responsible for speech comprehension, or Wernicke's area, located at the posterior end of the superior temporal gyrus and neighboring gyri of the parietal lobe. In order to connect these two important speech centers, the arcuate fasciculus must pass around the insula and parallel to the superior longitudinal fasciculus. The cingulum bundle, or cingulate fasciculus, connects the frontal and parietal lobes with the parahippocampal gyrus and temporal lobe, passing through the white matter of the cingulate gyrus superior to the corpus callosum. And lastly, there is the inferior longitudinal fasciculus, or temporo-occipital bundle, that connects the temporal and occipital lobes with fibers running below the inferior horn of the lateral ventricle and lateral to the optic radiations. Good. Now that we've covered these fibers, let's make a coronal section through the brain and identify a few more white matter structures. As you can see here, between the putamen and the claustrum, there's a thin sheath of white matter called the external capsule. Its dorsal part contains the claustrocortical fibers, which run between the claustrum and the frontal and parietal lobes, while the ventral part contains fibers of the uncinate fasciculus and fronto-occipital fasciculus that we previously discussed. Also, more laterally, the claustrum and the cortex of the insula are connected by the fibers of the extreme capsule. Finally, we get to a structure called the internal capsule, which contains efferent and afferent projection fibers. Think of it as a highway of axons that signals both to and from the cerebral cortex, connecting it with more caudal structures like the thalamus, brainstem, and spinal cord. Let's make an axial section through the brain to visualize it better. As you can see, the internal capsule runs lateral to the thalamus and through the nuclei of the basal ganglia, separating them into the lentiform nucleus laterally and the caudate nucleus medially. The internal capsule has a V shape and consists of 5 parts: the anterior limb, genu, posterior limb, retrolentiform, and sublentiform parts. If we look at the anterior limb of the internal capsule, it is located lateral to the head of the caudate nucleus, medial to the lentiform nucleus, and contains 2 types of fibers, the corticopontine fibers and frontopontine fibers, that run from the frontal lobe to the nuclei of the pons, and the thalamocortical fibers that form the anterior thalamic radiation, originating from sensory relay nuclei of the thalamus and traveling to the frontal cortex. The genu is a flexure just medial to the tip of the lentiform nucleus and contains the corticobulbar fibers, which run from the cortex to the motor nuclei of the cranial nerves that control the movements of the head and neck. The posterior limb of the internal capsule is located lateral to the thalamus and medial to the lentiform nucleus, and contains the corticospinal fibers, which run from cortex to lower motor nuclei in the spinal cord that control the voluntary muscles of the trunk and extremities. The fibers of the corticospinal tract within the posterior limb are organized in a rostral to caudal fashion, with fibers controlling the upper extremities located rostrally, and those controlling the lower extremities located more caudally. The posterior limb also contains axons of the medial lemniscus, the spinothalamic tract, and also some thalamocortical fibers. These thalamocortical fibers form the superior thalamic radiation that spreads from the thalamic nuclei, processing sensory information towards the cortex of the parietal lobe. The retrolenticular part of the internal capsule is located posterior to the lentiform nuclei and contains the geniculocalcarine fibers, otherwise known as the optic radiation. The optic radiation originates from the lateral geniculate nucleus of the thalamus and spreads posteriorly, carrying visual system information to the primary visual cortex of the occipital lobe. Lastly, the sublenticular part of the internal capsule is located just inferior to the lentiform nucleus and contains the fibers of the auditory radiation, which originate from the medial geniculate nucleus of the thalamus and spread towards the primary audio cortex of the temporal lobe. Alright, as a quick recap. The white matter of the brain contains axons that connect the cerebral cortex with other parts of the nervous system. The connections include commissural, association, and projection fibers. Commissural fibers connect the left and right cerebral hemispheres and form the corpus callosum, the anterior commissure, posterior commissure, and the hippocampal commissure. The largest bundle of white matter is the corpus callosum, which sends signals between the 2 cerebral hemispheres and connects them together. The anterior commissure is a much smaller white matter structure that mainly connects the olfactory bulbs and the temporal lobes of each hemisphere. The posterior commissure is a small commissure located in the posterior diencephalon and contains crossing axons from the oculomotor complex. And lastly, the hippocampal commissure connects the left and right hippocampi. On the other hand, association fibers connect different cortical regions within the same hemisphere. These fibers include the short association fibers, which connect the cortices of the adjacent gyri, and the long association fibers that connect the cortices of distant gyri. Long association fibers include the superior and inferior longitudinal fasciculi, the fronto-occipital fasciculus, and the uncinate, arcuate, and cingulate fasciculus. Finally, the projection fibers are axons that send either ascending or descending signals and travel within the limbs of the internal capsule. These fibers connect with the cerebral cortex with other nervous system structures, like the thalamus, brainstem, and the spinal cord.